So good evening, everyone. A warm welcome to all of you who have participated in the Synapse 2021. It's a great pleasure to welcome you all to the second version of Synapse 2.0. We hoped last year that we could go on for an in-person meeting. Unfortunately, the pandemic has extended and we are still with the webinar. But nevertheless, the enthusiasm for participation has not stopped or hampered. So before we move on to today, just a couple of minutes on last year's event. So we started this effort on having Neuroscience Symposium short lecture series and symposiums for one day last year with interesting speakers from all over India and also from Harvard University. And this was a very good success. And we thought to move forward this year with 2 days, expanding it to one more day. So we have 2 days this time. And then we have 8 speakers. And we have included lightning talks this year. So we had enthusiastic participation for lightning talks as well. And so we shortlisted and we have 20 lightning talks, which will be judged by experts. So as you see here in my screen, we have excellent speakers for the sessions today and tomorrow. I request all of you to stay for all sessions and enjoy the talks. Prior to moving on to the talk sessions, I would like to welcome our director, ICER Tirupati Director, Professor K.N. Ganesh, to share a couple of thoughts on Synapse 2021 and to open the session. Professor Ganesh, warm welcome to you.
Yeah. Can you hear me?
Yes.
Okay. Yeah. First of all, let me, on behalf of ICER Tirupati, welcome you all for this Neuroscience Symposium, Synapse 2021. This is the second edition of the Neuroscience Symposium. Last year, it was organized by ICER Tirupati. And this year, very gladly, ICER Trivandrum has joined. And hopefully, as this proceeds further, more and more institutions will also join. I was told by Dr. Vasudha that the number of registrations are quite a high number, and it tells about the growing interest in this area. The neuroscience itself is very, very important because finally the aim is to understand the function of brain, the final goal. And synapses, as most of you know, are the sites of contact between the nerve cells, and they convert electrical signals into chemical information, which is conveyed between neurons at this site. We have around 100 billion nerve cells or neurons in our brain, which is perhaps more than all the stars in our galaxy. And a single neuron can be directly connected with at least 10,000 others. So this gives rise to a staggering 100 trillion more nerve connections. And if each of these neurons were laid end to end, I was told that this would perhaps wrap around the earth twice over. So that is the complexity of the neuroscience. And to understand this, certainly one has to use different kinds of techniques, ranging from electrophysiology to imaging at the fast time scales, etc. So it's a very fascinating area, and I'm glad that the second edition now is attracted quite a lot of things. And also, I must say that we have a very good speakers list. I have gone through that, a multi-institutional one from NCBS. We also have some speakers from abroad, and I'm sure they will all light you up with this as the talks proceed. And this is certainly online symposium, and I think rightly so. The organizers have kept just 2 hours or so, without without loading too much of information on everyone. This is the new way. Once the online meetings have become the routine, I think we can just have about 2 hours symposium. Whereas if you do in person, you spend a whole day, covering all the topics. So it's very good. So today we have a few talks, and tomorrow, and also I see from the program that there are flash talks, and also I think some prizes are given to some of the best talks. So with this very brief introduction, and I'm very excited about the area itself, the neuroscience. And Dr. Vasudha has been leading this program at IISc Tirupati, and I'm sure in times to come, we'll expand to include other institutions also in this meeting. With this very brief introduction, I welcome you all, and special warm welcome to all the speakers who have accepted to talk in this symposium. Welcome you all, and I'm sure there will be very good proceedings, and all of you will feel enriched by the end of the symposium. All the best for the symposium.
Thanks a lot, Professor Ganesh. That was really kind of you. Next, I would like to invite Director, IISc Thiruvananthapuram, Professor J.N. Murthy, to share a few thoughts with us. Professor Murthy.
Thank you. Thank you, Dr. Vasudha and Dr. Poonam. Very good afternoon. And I echo what Professor Ganesh said. A warm welcome to all the participants of this two-day symposium. Ideally, we would have really liked to have conducted this symposium in our beautiful campus. Given that the first version was held in IISc Tirupati, of course, online way, but we were looking forward to doing this symposium in our campus. Unfortunately, with the pandemic still continuing, that's not going to be reality for some more time. Otherwise, well, I was just looking at our School of Biology, and our School of Biology has a small number of relatively, in terms of the faculty strength, about 14. And if you look at the spread of the areas that are practiced, and we have starting from ecology to biochemistry, biophysics, molecular and cellular biology, and of course, the immunity and what is the, I guess immunity and infectious diseases, yes, I forget. And we have added a new area here, neurobiology. Right now, we have a lone faculty member, and which is how we see Dr. Poonam Thakur here. And I would like to tell all the participants that, in fact, we have made offers. We would like to expand this, and soon we'll have one more addition to Dr. Poonam Thakur. She joined our institute just a year and a half back, and I'm really glad that within a short time, she has gotten together with the professionals in the country, and getting together with IISc Tirupati, she has put together this program. I think Professor Ganesh mentioned about it, and in fact, Dr. Vasudha talked about it, and we have set up about eight talks. I think some of the names are even to a chemist like me who is very remotely connected to biology. I don't even have any overlap with biology. In fact, I work more on material science, but not even remotely with biology. Even to me, I think some names are very, very familiar in the area of biology. But speaking of neurobiology, I think to a chemist like me, I think Professor Ganesh is really well poised to talk about it, because he is more of a bioorganic chemist. And in fact, he gave a brief introduction to neuroscience, but I cannot do that. But to a kind of a naive person like me, moment to think about neuroscience is all about nerve cells, brain, and neural transmission. And of course, cognition comes into my mind too much, because I was the convener of educational policy committee for IISc Thiruvananthapuram for about, IIT Kanpur for about a year and a half. And which was when the idea of basically starting a new center on cognitive sciences was mooted, and I sort of took it forward. In that connection, I was really wondering, I think if you talk about cognitive sciences, which has come into being in its own right today, but functionally, if you look at it, I think everything boils down to neuroscience. I think it's a very great area, and I'm really glad that we'll have this particular area basically expanding in times to come. The symposium, it was talked about, has 8 speakers, punctuated by the so-called the lightning talks, And in fact, we speak of them as a flash presentations. And we experimented that in chemistry department, School of Chemistry here, giving about 3 to 4 minutes to the students. And it was really a grand success. But I see that here, every speaker has been allotted about 4 to 5 minutes, I suppose. That's a wonderful opportunity. I think, they are really great. In fact, allows the students to sort of, articulate well, think about what to present in a very concise manner to a broader audience, and to bring out in a very shortest time the best of what an individual has. I think this should be really encouraged. And towards the end, I note that there are also going to be prizes, which is really wonderful. I think, what do I have to say? I think I'm very sure, given the short but very effective speakers, the participants will carry home a very enriching experience. And I'm sure that the proceedings next 2 days, the version 2 has 2 days, which is what was talked about. And I'm sure they'll be great, and wish the Symposium a grand success. Thank you very much.
Thank you, Professor J.N. Murthy. Thank you so much for this, highlighting also the lightning talks. I thank both Professor K.N. Ganesh and Professor J.N. Murthy in terms of the point that they also made that, lightning talks are interesting. And that's what we actually wanted to bring in as a new flavor, wherein our PhD and postdoctoral people can come together and showcase their work, and also in this way network essentially, because that's somehow missing. Neuroscientists, neurobiologists in our country are placed here and there, and we wanted to utilize this opportunity to bring all these young people, upcoming postdocs and so on. So thank you again. And also I'd like to acknowledge at this stage that the prizes that the directors rightly mentioned were sponsored or are sponsored by Author Cafe, the TNQ publishers in Chennai, and very glad that they were coming up to do this, and this is very nice. And so that sort of motivates also to the students to give their best essentially. And at this stage, our chair, Professor Ramesh Sonti, is not available, he's not there, but I'd like to extend also the motivation and thanks to both Professor Ramesh Sonti and Professor B.J. Rao, who was instrumental and helped us start this whole thing last year. So both of them couldn't join, but that's fine. They'll join eventually during the talks. So before we move on to the session, this is the schedule for today. So I'll have a couple of important instructions, housekeeping instructions. Please, all of you, keep yourself muted during the talk, except the speaker, and except the speaker, others would be kept muted. And with the first session, we have Dr. Vladimir Sitnik from University of New South Wales, Sydney, and then followed by that is Professor Shubha Tole from TFR Mumbai. Then we'll have a short break. We'll move on to lightning talks. Once the lightning talks are over, we'll have 2 other talks. And since we had overwhelming registration, we are also live streaming this event, and we have shared with all of you the live streaming link also. If you did not get a place, if your friends did not get a place, we apologize. Please redirect them to the YouTube link, and that would be really helpful. So, and then if you have questions, you can please put it up in the chat box. We will moderate the questions. If there is some time left, you can raise your hands and we may invite you to ask live questions. So during the talk, you can feel free to post the questions in the chat box. All right, so with that, we are ready to move to the first talk for today, the first talk of the first session. Let's get started. The talk by Dr. Vladimir Sitnik. He's an associate professor at the University of New South Wales in Sydney, Australia. His lab uses a number of biological systems ranging from biochemistry to electrophysiological methods, and they study systematically from cellular to tissue to organ level understanding, and they address important issue of how neurite outgrowth or synaptogenesis neurotransmitter release and synaptic plasticity takes place in the brain. So before to this position, Dr. Vladimir Sitnik was in Germany in the Center for Molecular Neuroscience, Hamburg. And today's topic, he's going to take us through the function of NCAM2 in regulating the function of beta amyloid precursor protein enzyme 1. With this, I invite Dr. Vladimir Sitnik to share his screen and deliver his lecture. Welcome, Vladimir Sitnik.
Thank you, Vasudha. Yeah, so I will try to share my screen. All right. So
we can see your screen, yeah, and
yeah,
you're
perfect.
audible.
Yeah, thank you. Can you see my pointer, my arrow?
Yeah.
Yeah, great. Well, first of all, I would like to thank the organizers for this great opportunity to present our work, and it's great to hear that there is so much interest in neurobiology. And yeah, so this picture just to show where our lab is located. So we are located in Sydney, in the University of New South Wales, and my lab is somewhere here at the end of the campus and closer to the ocean. As Vasudha mentioned, we are interested in neural cell adhesion molecules, and today we'll show you some of our published work and some of our new work about the neural cell adhesion molecule 2. Neural cell adhesion molecule 2 is the large protein which has a very large extracellular domain. It is an integral membrane protein. It has a transmembrane domain and a short intracellular domain. It is a member of the immunoglobulin superfamily because it has 5 immunoglobulin domains or repeats in its extracellular domain. It also has 2 fibronectin type 3 domains in structure. So the extracellular part of NCAM2 is important because it can bind to itself, or more precisely, 2 NCAM2 molecules on membranes of 2 cells can bind to each other, and thereby they form so-called adhesive bonds. So these adhesive bonds connect membranes of these 2 cells, and they're mostly mediated by immunoglobulin domains, so which can bind to each other. So this picture shows you a more detailed structure of NCAM2, and in this model, it is proposed that the homophilic formation of homophilic adhesive bonds is actually mediated by first 2 immunoglobulin domains in NCAM2 structure. And this picture also shows you that NCAM2 is a glycoprotein because it has a lot of these carbohydrates which the extracellular domain of NCAM2 carries, and which can also modulate the formation of homophilic adhesive bonds between 2 molecules. When we started working on NCAM2 was not really well characterized, and so this is one of the first pictures we obtained using our confocal microscope, and it shows you the distribution of NCAM2 in the cultured hippocampal neurons from a mouse. You can see here that NCAM2 is expressed in these neurons. It is present in cell body. It is also present along dendrites, which are also positive for MAP2, a marker of dendrites. And you see it sometimes is found in these small clusters. And if you look now at high magnification and look at the position of synapses, which are marked with the antibodies against synaptophysin, you can see that these small clusters of NCAM2 colocalize with the accumulations of synaptophysin, which tell us that NCAM2 is a synaptic adhesion molecule because it accumulates in synapses. We can also label neurons for a postsynaptic marker protein, PSD95. So this protein accumulates in the postsynaptic part of the synapse in dendrites. And you can see that clusters of NCAM2 also colocalize with clusters of PSD95 And these accumulations of NCAM2 and PSD95 are opposed to accumulations of synaptophysin. So this tells us that NCAM2 really sits in the postsynaptic density in synapses in these neurons. Before we worked on NCAM2, we also worked on a number of other adhesion molecules. I know there are other labs who worked on other synaptic adhesion molecules. And this slide is just to illustrate what is known about the functions of synaptic adhesion molecules. On this slide, you can see this is a presynaptic part, axonal part, where neurotransmitters are stored in synaptic vesicles, and these vesicles fuse with the plasma membrane and release neurotransmitters. And this part is a postsynaptic part. It's a dendritic part where neurotransmitter receptors are located, which detect neurotransmitters released from the presynaptic part. And these guys in red here are synaptic adhesion molecules. So if you look carefully at this picture, you can see that these are the only proteins which connect the presynaptic part to the postsynaptic part. So one of the important functions these proteins play is to mechanically connect axons to dendrites of neurons. In addition, what they do, they bind to scaffolding proteins such as PSD95 or spectrin. And via scaffolding proteins, they're linked and recruit other proteins to synapses, such as neurotransmitter receptors. And in addition, what they do, they align the presynaptic part with the postsynaptic part, or they align the presynaptic components with the postsynaptic components. They determine where vesicles fuse with the plasma membrane and release neurotransmitter, and they recruit postsynaptic receptors to this location so that they can efficiently detect neurotransmitters when they are released. We've got interested in NCAM2 because of the genetic association between NCAM2 and human disorders, and we were particularly interested in Alzheimer's disease. And there were several studies which showed potential association between NCAM2 and this disorder. First of all, NCAM2 is encoded by a gene which is located on chromosome 21 in humans. And chromosome 21 in humans, when it is duplicated, it causes Down syndrome. And what we know about Down syndrome is that all patients with Down syndrome inevitably develop Alzheimer's disease. So this is obviously an interesting connection, but in Down syndrome, many other genes are triplicated, so it's not very direct. However, there were other studies, for example, this one shown here, showing that variations in NCAM2 gene is a genetic factor which is related to the progression of Alzheimer's disease. And there was another very interesting study showing that not only NCAM2 is a genetic factor related to the progression of Alzheimer's disease, but intronic single nucleotide polymorphism in NCAM2 gene are associated with the increased levels of A-beta even in controls, in still healthy individuals. So there was this interesting relationship between NCAM2 gene and production of A-beta. But the mechanisms were not known at all. And that's why we asked the question, so what could be the relationship? So what could be the mechanism? And to start this study, we collaborated with the Sydney Brain Bank, and they have a very large collection of postmortem human brain tissues. And we obtained tissues of individuals with Alzheimer's disease and age and sex match controls. And first, what we did analysis of NCAM2 levels in brain homogenease from those tissues. We analyzed several brain regions. We analyzed hippocampus. This is the brain region which is strongly affected in Alzheimer's disease. And for comparison, we looked at cerebellum and temporal cortex, which are less affected in this disease. And these graphs show you the results of Western blot analysis. Each dot here represent one individual from our collection, and you can see here on this graph that what we found was that levels of NCAM2 were increased in the hippocampus of people with Alzheimer's disease. Alzheimer's disease is very well known to be associated with the neuronal loss and also with the synapse loss. And this you can see on this graph showing levels of BACE1, a marker of excitatory synapses in brains of patients with the brains of people with the Alzheimer's disease. So you can see that levels of BACE1 are dramatically decreased in Alzheimer's disease, indicating a loss of synapses in this disorder. And this graph shows you levels of a marker of inhibitory synapses, VGET, and you can see that levels of VGET are not changed in AD. So the results were very interesting, but somewhat puzzling because, as I showed you before, NCAM2 is a synaptic adhesion molecule which accumulates in synapses. And the second graph shows us that synapses are lost. So then why do we have an increase in NCAM2 levels in AD? And is it really synaptic NCAM2 that we see here? So as an integral membrane protein, we know that NCAM2 is present not only in synapses, but can be also located outside of synapses. So we realized we need to look more carefully and analyze this tissue using other methods. And we used a method which we very often use to analyze tissue from our transgenic mice. So we prepared synaptosomes. Synaptosomes are particles which are formed by synaptic terminals when brain tissue is homogenized. So when we homogenize tissue membrane, so this is a synapse, you can see this axonal terminal, and in blue, dendritic terminal is shown, a spine. And when tissue is homogenized, membranes are broken around synaptic terminals. So they're broken somewhere here and here and here. And then they're sealed by lipids. This results in formation of these particles, which is outlined here in red. And then we can take this material, we can centrifuge it in the sucrose gradient, and these particles, because they differ in their density, they float. And we can collect them at the interface between sucrose and statin material. And then we can use this material and run a Western blot analysis. And this Western blot shows you that NCAM2 is also enriched in synapses in the human tissue. So here you can see synaptosomes from several brain regions from the human brain, from the cerebellum, cortex, and the hippocampus. And you can see that NCAM2 is particularly strongly enriched in synapses from the hippocampus. And this is synaptophysin is a marker of synapses showing you that the preparation was efficient and pure. Then we run the same analysis for our controls and people with Alzheimer's disease. And what is nice about this method is that we purify all synapses which remain in the tissue. So essentially we analyze really synapses which are still intact in the tissue. Here at the top you see an analysis of a control case. And again, you see that NCAM2 is highly enriched in synaptosomes from the hippocampus. And here you can see labeling for other synaptic proteins, for synaptophysin, PSD95, actin, B-glut. You see that they're all enriched in synaptosomes. And you see also that NCAM2 is enriched in synaptosomes from other brain regions, for example, from temporal cortex and cerebellum, just to a slightly lower degree than the hippocampus. And here at the bottom, you see synaptosomes from Alzheimer's disease patient. And you see that they still contain all synaptic proteins. So they contain synaptophysin, they contain a lot of PSD95, actin, B-glut. They're all still there. But if you look at the levels of NCAM2, you can see that levels of NCAM2 are dramatically reduced. And this is quantified here. So you see that first of all, NCAM2 is enriched in synaptosomes from the hippocampus to a much higher degree compared to other brain regions. And this enrichment is is lost, whereas other synaptic proteins are still quite enriched in these synapses. And this is despite the fact that levels of NCAM2 are increased in the hippocampus. So altogether, this suggested to us that probably the loss of NCAM2 is just the first step, which may lead to somehow lead to the synapse loss. So we also were interested in the mechanism, and what we did, we analyzed levels of NCAM2 in the soluble fraction. And we did this because NCAM2 is normally a membrane-bound molecule. So it's a transmembrane protein, which is an integral membrane protein. However, when it is cleaved, then this results in the release of the extracellular domain of NCAM2. And this extracellular domain then can be collected in the soluble protein fraction. And this analysis is shown here, where we analyzed synaptosomes and soluble protein fractions from control cases and from cases with Alzheimer's disease. And again, you can see that NCAM2 is highly enriched in the hippocampus in control cases, and it is present in the soluble fraction at a slightly lower molecular weight. And this slight difference in the molecular weight tells us that nearly the whole extracellular domain of NCAM2 was released to the soluble fraction. And if you now look in AD cases, you can see that the levels of the soluble NCAM2 are strongly increased in AD cases, whereas levels of the full-length synaptic NCAM2 are reduced. And this is also quantified here. So this tells us that the loss of NCAM2 in synapses is caused by its cleavage. We now know the reasons for this because we found that NCAM2 can interact with a beta peptide. Amyloid beta peptide is a toxic peptide which accumulates in brains of patients with Alzheimer's disease. And it is known to bind to a number of cell surface receptors. So NCAM2 is another one it can bind to. And this slide shows you that it binds in a concentration-dependent manner to purified NCAM2. And then to understand what this binding means and what it causes, we used, again, cultured hippocampal neurons. And we also, for comparison, analyzed cultured cortical neurons. And these Western plots show you homogeneity of those cultured neurons and synaptosomes prepared from cultured neurons. And you see that normally NCAM2 is highly enriched in synaptosomes from the hippocampal neurons and also is enriched in synapses, synaptosomes from cortical neurons. And then what we've done, we treated these neurons with A-beta oligomers, which accumulate similar to those which accumulate in the brains of patients with Alzheimer's disease. And you can see that this synaptic accumulation of NCAM2 was dramatically reduced in neurons treated with A-beta. This is quantified here. Surprisingly, we found that this effect was specific to the hippocampal neurons because we didn't see this in cortical neurons. So in fact, we found even an increase in NCAM2 levels in cortical neurons treated with A-beta. We are still working on the mechanism and trying to understand why this is happening. So I cannot answer you today what is the mechanism, but this was really surprising, but nicely correlating with our Western blot data showing that NCAM2 is affected only in the hippocampus of AD patients, and also nicely correlates with the fact that the hippocampus is the most affected area in AD, whereas the cortex is much less affected in Alzheimer's disease. The next question we asked was whether this loss of NCAM2 is actually causing problems, whether it can affect the synapse integrity. And to answer this question, we decided to acutely disrupt homophilic adhesive bonds which are formed by NCAM2 molecules. So on this picture, you can see this homophilic bond, which is formed by two NCAM2 molecules which sit on membranes of presynaptic part and postsynaptic part. So what we did, we took a soluble fragment of NCAM2, the whole extracellular domain of NCAM2, and we reasoned that it will compete for binding in this adhesive bond with membrane-bound NCAM2 and disrupt adhesive bonds as shown here. We also tested an antibody against the extracellular domain of NCAM2 because antibodies are very large proteins, and when they bind to their targets, they sterically interfere with the interactions of the proteins they bind to. So we decided to use this system, and again, we used cultured hippocampal neurons, and here at the top you can see control neurons with intact homophilic adhesive bonds. And you see here that these neurons were labeled for a presynaptic marker protein synaptophysin and postsynaptic marker protein cell surface GLUR1, which is a subunit of AMPA receptors. And you see that the presynaptic and postsynaptic signals nicely colocalize, indicating alignment of presynaptic and postsynaptic parts in this neuron. And here on these panels, you see what happens after we incubated neurons with the soluble extracellular domain of NCAM2. You see that this alignment, which we see in the control neurons, is largely lost because we see a lot of these green clusters of cell surface AMPA receptors, which are not anymore colocalized with the red clusters of synaptophysin. So essentially, when we disrupted these adhesive bonds, we induced this mislocalization of presynaptic and postsynaptic components. And also we found a drop in the intensity of synaptophysin along dendrites of these neurons, indicating that some of the synapses were lost. And the same effect we found in neurons which were treated with NCAM2 antibodies, indicating to us that it was really due to disruption of adhesive bonds between formed by NCAM2 rather than binding of the soluble extracellular domain of NCAM2 to some other receptors, which is still possible, but altogether this data suggests that this was really caused by disruption of NCAM2 adhesive bonds. And what was interesting, that a similar effect we found in neurons treated with A-beta peptide or A-beta peptide oligomers, which is known to cause synapse loss and also misalignment of the presynaptic and postsynaptic components. And this data is quantified here. So you see that in neurons treated with the soluble extracellular domain of NCAM2 antibodies against the extracellular domain of NCAM2 or A-beta, there is an increase in non-synaptic AMPA receptors, and there is a loss of synaptophysin immunoreactivity along dendrites of these neurons, indicating synapse loss. So this was interesting. So we data were published in our paper in Nature Communications. However, we didn't answer all questions we had because we still were puzzled by this relationship between NCAM2 and high A-beta levels. So in my second part, I will talk about our still unpublished data. So it's currently under review. I hope it will be published soon, but it's still not published, so I hope it will be interesting. What we know about A-beta is that it is produced from amyloid precursor protein. Amyloid precursor protein is this transmembrane protein, and it must be cleaved at two sites to generate A-beta. And the first cleavage, which is also a rate limiting step in this cascade, is mediated by BACE1, this beta site amyloid precursor protein cleaving enzyme 1. So BACE1 cleaves APP slightly above the plasma membrane, and then the second cleavage is done by gamma secretase, which cleaves APP within the membrane, and this causes formation of this A-beta peptide shown here in red. I hope you can see here that BACE1 is also transmembrane protein, and it has this large extracellular domain, which is normally, however, is located in the lumen of vesicles, because a lot of BACE1 is located inside of endosomes, which confuse with the cell surface, and then this domain becomes a cell surface localized domain of the protein. So we first ask the question whether BACE1 can cleave NCAM2, and perhaps this can be the reason why NCAM2 is lost in synapses. And to answer this question, we prepared this construct shown here on the left. It coded for NCAM2 with an HA tag at the end terminus of the protein. So we can detect cell surface NCAM2 with an antibody against the HA tag. And we also used an antibody against the C terminus of NCAM2, which bound to NCAM2 somewhere here. And you can see that in these cells, these are CHO cells transfected with this protein and labeled with antibodies against HA tag and NCAM2 C terminus tag. You see that the signals colocalize, and we have plenty of NCAM2 at the cell surface. And this panel shows you labeling of cells which were co-transfected with this protein plus BACE1. And you see that almost all labeling for HA tag is lost in these cells, so indicating that this part of the protein was lost. What was interesting, however, was that this cell retained some of the NCAM2 C-terminus immunoreactivity. And this is quantified here. We see that in cells co-transfected with BACE1, there was a reduction in the ratio of NCAM2 HA tag immunoreactivity relative to its C-terminus immunoreactivity, indicating cleavage of NCAM2. And we found a similar effect in cells co-transfected with BACE2, another isoform of BACE, which also cleaves NCAM2 according to this data. So we were quite happy about this data. And then, as it often happens in science, there was a paper published in Journal of Biological Chemistry showing what I just showed you, that BACE1 cleaves NCAM2, and this was shown in vivo. So we were on the one hand upset, but on the other hand, we were quite happy that our results were reproduced, or another lab obtained similar results to our results, showing that NCAM2 can be cleaved by BACE1. Because we use this assay, we have noticed a very interesting thing. We found that in cells which were co-transfected with the BACE1, NCAM2 immunoreactivity was often found in such small clusters. And when we compared this labeling to the labeling with the antibodies against BACE1 or antibodies against flag tag, which was used in this case, we found that these clusters often overlapped, suggesting to us that not only NCAM2 C-terminus immunoreactivity was retained, but it colocalized with BACE1. And we then checked in whether we can find a similar effect in neurons, and indeed we found that in neurons, in dendrites, we often found these very bright spots containing a lot of NCAM2 C-terminus immunoreactivity. And these spots, hot spots of NCAM2, colocalized with the accumulations of BACE1. They were often found in dendrites, in dendritic shafts, but also sometimes in spines, as you can see here. And NCAM2 not only colocalized with BACE1, but they interacted, because when we did a proximity ligation assay with the antibodies against the C-terminus of NCAM2 and C-terminus of BACE1, we found that these two proteins were close to each other enough to induce a proximity ligation reaction. So that you can see these proximity ligation products here. They were often found in cell bodies of neurons and along dendrites, and also close to synapses in these neurons. To confirm this, we also use another assay, which is called biomolecular fluorescence complementation assay. And in this assay, what we've done, we've taken just a fragment of NCAM2 comprising the transmembrane and intracellular domain of NCAM2. We've used this fragment to a portion of the fluorescent protein Venus. And we've obtained, via collaboration, a construct coding for BACE1 fused to fused at its C-terminus to another portion of Venus. And what we know is that when these two proteins interact and these two fragments are brought close to each other, they can recombine and reconstitute the fluorescent protein. And this is what happened. So when we co-transfected this fragment and BACE1, we found that the venous fluorescence was restored, and we can see a lot of green fluorescence in our cells, indicating that this fragment and BACE1 can bind to each other. And we found this not only in CHO cells, which we used to start with, but also in cultured hippocampal neurons. And the BFIC signals were found in dendrites. You can see the small dots. They're often found close in spines or near synapses, for example, in this location. And they're also found in axons of these neurons. For example, here you can see these accumulations of BFIC which overlap with synaptophysin. So the question was what these complexes do. If you look at the distribution of NCAM2 and BACE1 in brain tissue, you can see that, for example, in hippocampus, NCAM2 C-terminus or NCAM2 aminoreactivity is enriched in dendrites of neurons. So this region here is called the dentate gyrus. It contains granule cells, and their dendrites are located in this layer. So this is where NCAM2 particularly accumulates. And if you look at the distribution of BACE1, it is very well known that it accumulates here in this structure, which is called the mossy fibers. And these mossy fibers are formed by axons of granule cells, which dendrites of which are enriched in NCAM2. So you can see this colocalization of NCAM2 and BACE1 in dendrites of granule cells, but not so much in mossy fibers. And what we also know about BACE1 is that it says this protein is made in cell bodies, transported to dendrites, and then targeted to axons. And this process is called transcytosis. So we ask whether NCAM2 plays any role in this process. So first we looked at the levels of BACE1 at the cell surface of NCAM2 knockout neurons, and we found that levels of BACE1, I hope you can see this on this image, are higher at the cell surface of NCAM2 knockout neurons compared to wild type. And then we analyzed that high magnification dendrites of these neurons, and we found that as it is known that a lot of BACE1 is present in intracellular accumulations, which are seen here in red, but in knockout, the number of such accumulations was very much reduced. So most of BACE1 was stuck at the cell surface of NCAM2 knockout neurons. And then we did an experiment with the CHO cells, which were co-transfected with the NCAM2 and BACE1, and we analyzed distribution of BACE1. And we found that in control cells, BACE1 was not so much enriched in Ldb1 positive endosomes, but when we co-transfected cells with NCAM2, a lot of BACE1 was now found in this Ldb1 positive endosomes, and this way it colocalized with NCAM2. And a similar effect was found when we co-transfected cells with this fragment comprising the transmembrane and cytoplasmic domain of NCAM2. This is quantified here. So this all suggested to us that NCAM2 or its fragment can target BACE1 to Ldb1 positive endosomes, which are known to be involved in transcytosis. So to confirm that NCAM2 can trigger transcytosis of BACE1, we analyzed slices of brain tissue, and we analyzed BACE1 levels in the mossy fibers. And what we found that BACE1 levels in the mossy fibers, this region is called stratum lucidum, were reduced in NCAM2 knockout mice. Interestingly, we found that levels of NCAM2 were also reduced in molecular layer where dendrites of granule cells are located. But even if you take into account this reduction and take a ratio of BACE1 levels in stratum lucidum to the molecular layer, we still see a significant decrease in this ratio, indicating reduced targeting of BACE1 to synaptic terminals. And this is high magnification of synaptic terminals of mossy fiber.
Volodya, sorry to interrupt. You have last 4 minutes, then we can have time for question. Yeah.
Right. I'll try to finish quickly.
Thank you.
Yeah, thanks. Okay, so does it affect the activity of BACE1? And the answer is yes. So we analyzed the substrate cleavage of BACE1 substrates, and we found that BACE1 that NCAM2 knockout mice show a significant reduction in cleavage of SZ6, which is a bona fide substrate of BACE1 and CHL1. And we also found pathfinding errors in these animals, which are known to be present in BACE1 knockout mice. The loss of BACE1 was due to cleavage of BACE1. So this plot shows you levels of BACE1 in homogenates of mice. And we'll probably go quickly through it, but you can see this increase in soluble BACE1 levels in the soluble protein fraction indicating shedding of BACE1. And it was increased in knockout mice. And the last two slides to show you, so what we think these mechanisms, what is the role of this mechanism in control neurons? And what we did, we analyzed whether this mechanism can be triggered by synaptic activity. And the reason was because we found that NCAM2 is enriched in synapses. So what we did, we induced chemical long-term potentiation of cultured hippocampal neurons. And here you can see control neuron. We see that BACE1 is present in axon of this neuron, which is negative for MAP2. And then when we induce chemical long-term potentiation, we see this increase in NCAM2 in the BACE1 levels in the axon. And it is transient because 60 minutes later, it is lost. And you can see this summarized here, but in NCAM2 knockout neurons, this mechanism is totally blocked. So there is no increase in BACE1 levels in axons in response to chemical long-term potentiation. BACE1 deficiency is known to induce problems with working memory, and we used a test which can analyze working memory in mice. And this test is called Y-maze. So in this test, you can put a mouse in an apparatus which looks like a big Y letter. You can block one of the arms, and the mouse can explore just arm B and start in arm A. And then you do a test when you remove this door, and the mouse can explore all arms. But if the mouse remembers arm B, what it does, it explores more often novel arm C. So this is a test of short-term memory because the mouse must remember arm B to have preference to arm C. And this is analysis of our NCAM2 knockout mice showing that wild-type mice can remember arm B and arm A because they show preference to arm C. Heterozygous animals can still remember, but in knockouts, this preference is totally lost. And they spend less time in the novel arm, and they travel shorter distances in novel arm. So this is the summary. So we think what happens in dendrites, BACE1 cleaves NCAM2, this cleavage targets BACE1 to RAB11 positive endosomes, and they travel from dendrite to the axon to the terminals where BACE1 can cleave its substrate involved in remodeling of synaptic terminals. And this coupling between postsynaptic remodeling and presynaptic remodeling is potentiated by long-term potentiation. And this mechanism is blocked in knockout, and BACE1 accumulates at the cell surface of dendrites and it's shed, and that's why its activity is lost. And this causes problems with the formation of the muscle-fiber structures in brains of these mice, and also causes problems with short-term memory. Okay, and I would like to thank all people who were involved in this work and helped us to do all these experiments. First of all, my students and also Irina and Ryan, Shangfeng, Sarosh, Grant, Irina, they did a lot of work on analyzing animals, immunohistochemistry, immunocytochemistry. Kelly and Devanavan, they helped with the behavioral analysis of mice. Jenny helped with the SZ6 work. Melita helped with our CHL1 work. And Glenda and Claire, they helped us with the analysis of human brain tissue. And obviously, I would like to thank our funding bodies. And thank you all for listening to this talk.
Thank you, Volodya. That was a really wonderful and intriguing talk, particularly because you spoke so much about synapses, and this being Synapse 2021, it fit well. So I think we have time for one or two questions, although there are several questions in the chat box. So Prakash Devaraju asks if there is any age dependency of NCAM-mediated phenotypes.
Yeah, that's a very good question. So we know that young mice also show deficits, and what is known that loss of NCAM2 is found in people with the neurodevelopmental disorders, and that's probably why its role in Alzheimer's disease is not really well studied, because when NCAM2 gene is lost, people simply develop neurodevelopmental disorders. We are still analyzing older animals. We don't know what loss of NCAM2 causes in really old animals. So, but we know that loss of NCAM2 in really young animals also causes deficits related to autism.
Okay, thank you. So Mallikarjuna asks the impact of beta monomer to beta fibril formation on NCAM2 and synapses in hippocampus.
What we know is that oligomers of A beta bind to NCAM2 rather than monomers. So this formation of small oligomers is the trigger which causes loss of NCAM2. Whether monomers of A beta bind, so we do not detect them when they interact, but it's very well known that only oligomers are the most toxic species which cause synapse loss. Yeah.
Okay, so there's a question from Pranay B. Gaikwad. So he would like to know that that the cell adhesion molecule family, they have two NCAMs, NCAM1 and NCAM2. And he wants to know if the NCAM1 is vital protein involved in synaptogenesis and synapse maintenance. So he's asking, what about NCAM1?
NCAM1 is also cleaved by BACE1, and we do have some data. So it was already known that NCAM1 is also affected in Alzheimer's disease. In contrast to NCAM2, it's probably more broadly affected. So we were really, surprised to find this very specific targeting of NCAM2 only in the hippocampus. So we didn't see this for NCAM1.
Okay. So I'll take one last question. Maybe after this, Valodia, you could answer some of them. Sangeeta Shri is asking about recent studies on dissolution of amyloid beta through various strategies. So she'd like to know if we somehow succeed in dissolving A beta in AD patients, what would be the fate of neuronal transmembrane proteins? Will they get to normal state, or how would it be? She's asking this.
Yeah, that's a very good question. Yeah, so I think we need to do more research on this, because what is important is the balance, because we don't want to create more oligomers because they're more toxic. And when we dissolve larger oligomers, we can create more smaller ones, which can still be breaking adhesive bonds and causing cleavage of NCAM2. So it should be really either a strategy which rather clear all oligomers, or really causes complete dissolution of oligomers, which I'm not sure whether it can be achieved. But I think we just need to do more research on this and check this.
All right. Thank you. So there are a few more technical questions also in the chat box. If you can, if you have time, I know it's too late for you already there in Australia, maybe you can answer them. And yeah, with this, we can move on to the next speaker, which is Shubha Thole. Just a second. Yeah. So I hope you can see my screen. So yeah. So she probably doesn't really need introduction to the to all of us, but still I'll try to say a few words. She's Senior Professor and Dean of Graduate Studies at TIFR Mumbai. Her lab worked in the field of forebrain development with a focus on self-fate determination in hippocampus, like we know from her title, how to make a hippocampus. So they are interested in neuronal subtype specification in the neocortex and control of axon pathfinding and innervation of the cortex. Professor Thole is a recipient of several awards, including the Infosys Science Foundation Award, the Shanti Swarup Bhatnagar Award, and the Research Award for Innovation in Neuroscience. So with this, I would like to welcome her to give the talk on how to make a hippocampus. Following the Synapse talk, we are really interested in learning how to make a hippocampus. Welcome, Professor Shubha Tole. Over to you. I stopped sharing and you can share the screen.
Hello.
Hi. Yeah, we can hear you. We can see you.
Perfect.
Yeah.
Great. So thank you very much for inviting me to this. I really appreciate the energy and enthusiasm put in by the organizers and the students, especially in these difficult times. To continue investment and engagement in science is so nice to see in this, the next gen, as it were. So I'm very happy to participate. And I would be very happy to be interrupted with questions. So it's hard for me to track them in the chat. So I don't mind if you just, start speaking, because it's let it be an interaction. Otherwise, it can just be a recorded talk also. Right?
I just will interrupt you. I think at 30 minutes, we will have a buzzer for you just to alert you.
Sure.
And then, 40 minutes, 35 minutes, if you wrap around, there may be more time to interaction. Yeah, thank you.
Perfectly fine. Perfectly fine. Okay. All right. So here we go. There. Okay. My screen is all clear. All text is there.
Yes.
All right. Okay. So I'm going to share with you some. Can I request if you could spotlight my video, because I often show things with my hands, so it's useful for.
Yeah, I think IT will take care of this. We'll do this once again.
Okay. I'm going to share with you some insights we've had about the specification, the formation and the specification of the hippocampus using the mouse as a model. And I will end with a little data about human organoids, human embryonic stem cell derived organoids. This is our beautiful campus by the Arabian Sea. I guess I don't need to extol the virtues of the seacoast to you folks, but it's lovely. Would love to have people visit in person whenever the situation permits. Okay. Here is a cartoon of the mouse embryo at approximately embryonic 12.5 of a 19.5-day gestation. So the neural tube has formed, the forebrain has formed, and here we're going to focus on how this thin sheet of proliferating neuroepithelium knows where to form the hippocampus. Okay, the hippocampus normally forms in this medial portion of the sheet. So if the sheet is curved in, the hippocampus would form here. And what I will show you is a very localized signaling center where my fingernails are. It's very localized signaling center, one that we call the cortical hem, okay, shown here to express Wnt2b. This signaling center acts as an organizer, like a lighthouse from which signals emanate and the responding tissue actually becomes hippocampus. That is the first segment of my talk. It's work published somewhat some time ago, but I use it here to set up the question. Okay. Transcription factor Lhx2, limb homeodomain gene Lhx2. Here is an mRNA in situ, and you can see how Lhx2 is expressed pretty intensely in the entire cortical primordium, which at some point early in development does not know whether it is supposed to become hippocampus or neocortex. And fact is, hippocampus develops on the medial side of this primordium and neocortex develops on the lateral side. Now, two structures flank the cortical primordium. This is the Wnt and BMP expressing hem. It's like a thin tissue at the end of the cortex. We call it the cortical hem. We named this structure when I was a postdoc with Liz Grove at the University of Chicago. And at the other end of the cortical primordium is a DBX1 expressing structure called the antihem. And now these look like tiny structures, but you can see that this is a section. So if you look at the whole brain, if you glue many, many sections together, you can see the hem and the antihem are literally long strips of tissue, the hem being where the fingernails are in my hand model. Right? So it's like the hem and the antihem flank the cortical primordium. They are not cortical in that they don't produce cortical neurons. They produce Cajal-Retzius cells, which line the top of the cortex, but they don't produce projection neurons of the cortex. Okay. Why do I belabor this point about the hem and the antihem? Well, that's because of our early studies that showed that Lhx2 is a kind of a selector gene for cortical fate. In the absence of Lhx2, in the Lhx2 null mouse, there is no cortical primordium. And in fact, there is a great big expanded hem and antihem. Okay, it is not the case that the cortex died in the absence of LHX2. It could have been a survival factor. It's neither the case that the hem and the antihem overproliferated. But in fact, we showed, and I'm going to show you a little bit of the data, or at least a cartoon summarizing the data, that the cortex normally forms by suppressing hem and antihem fates. And the suppression of hem and antihem fates is done by LHX2. If you remove LHX2, the available progenitors become hem or antihem. Okay, we showed this by making a chimeric mouse, a mosaic, in which LHX2 null cells were embedded in a wild type mouse. And we showed that LHX2 null clusters on one side become hem and the other side become anti-hem. And we used this to see what happens when extra hems are embedded in the medial primordium. So what does an ectopic hem do? This kind of experiment was not possible to do by transplantation. These things happen very early in the embryo. But we could do it genetically by making this mosaic animal, which had ectopic hems. And to summarize all data, this is a control chimera in which all the green cells are just wild type cells, which happen to be derived from a GFP expressing embryonic stem cell line. And you can see that this chimera making does not affect the patterning of the tissue. The cortical primordium continues to express bona fide marker EMX1, and the hem continues to express Wnt2b. Now here is a wild type null chimera, where the green cells are all LHX2 null, derived from an LHX2 null embryonic stem cell line. And you can see how every green patch, okay, look at this rectangle, this triangle, or even this thin row of cells. Every green patch, I'm going to overlay these two on each other because they are the same section. See, every green patch actually becomes hem. And what I won't show you is that laterally, every green patch becomes anti-hem. So therefore, we could conclude that LHX2 is required for cortical fate. And by complement, loss of LHX2 on the medial side produces these ectopic hems. And then we examined what happens. Okay, what happens to the wild type tissue around them? See, what the ectopic hem can do could not be asked in a standard LHX2 null mouse because everybody has become either hem or anti-hem. There's nobody left to respond to these signals. Okay, but only in this chimera did we have wild type tissue available to ask, well, what do you become now that you're jammed next to a hem that you would normally never have seen? And the answer is that they become hippocampus. Here's a control. By embryonic 15.5, the hem shrivels away to the location of the fimbria, and you can see this little baby hippocampus here expressing PROX1, a dentate granule cell marker. And you can see in the chimeric mouse, each GFP patch expresses Wnt2b, signs of ectopic hem, and now you can see multiple PROX1 patches. And we've also shown multiple CA3 patches, multiple CA1 patches, and in fact, in a little older mouse, this is embryonic 17.5. Ignore this staining, which is in the thalamus. Look at LHX9 expression in the dentate and field CA3 in a control. And here is the chimera with multiple hems. And you can see in this hemisphere, so again, ignore LHX9 in the thalamus, but you can see 1, 2, 3. You can see several hippocampi, which are not necessarily all aligned because we do not are not able to control where the hem forms. So the hippocampi may get induced in any orientation, this being the C-shaped hippocampus on the dentate. And when you cut, you see these extra hippocampal patches. But note that between the dentate and CA3 patch, there is a negative patch here, which we showed upregulate CA1. So we get a nicely ordered hippocampal induction. So using this kind of approach, we were able to conclude that the hem indeed does act as an organizer for the hippocampus. So the answer to how to make a hippocampus is at least partially, here's a nice example from a different hemisphere. You can see two dentate gyri sort of packed one on top of the other, two V-shaped nested. Okay, now before anybody asks me, well, have I done behavior tests on these and do they have more memory? Let me pre-answer that saying we would have loved to test this. But for scientific and technical reasons, this was not possible. Scientifically, we do not think that the hippocampal fate is sort of specified but over-proliferated, and that's why you get more hippocampi. We actually think that an extra hem simply converts available tissue nearby to hippocampus when it should instead have become some other part of cortex, cingulate or retrosplenial cortex. So when we have an animal with extra hippocampi, we're actually losing some other modality. And this is impossible to control for. Okay? Technically speaking, we While the role of LHX2 in suppressing hem fate is cell autonomous, in that only null cells become hem, LHX2 is also required for erythropoiesis in the liver. So the null animals actually die at embryonic 15.5 because the livers are pale, they cannot produce red blood cells. And the chimeras die by E17. We suspect that it may be a non-autonomous requirement of LHX2 in the liver. So we could not get live births of these animals. So that sort of addresses the question of well, are these hippocampi functional? But the overall point is indeed, it looks like the organizer principle is at play here. The hem is the hippocampal organizer. Hem fate is normally suppressed by LHX2. I might have summarized this. LHX2 suppresses hem fate. LHX2 null clusters form hem only medially, and the hem is an organizer for the hippocampus. So this sets up the early portion of this talk to ask, well, if the hippocampus forms where the hem forms, then it simply pushes back the question, well, why does the hem form medially then? We have had some progress in answering that. We've discovered that an interaction between two other transcription factors, PAX6 and FOXG1, positions the hem medially. This is the work of Geeta Godbole, a postdoc, whose photograph I may have on this slide. He come up. Geeta showed that PAX6 by itself doesn't suppress hem fate. If you remove PAX6, you don't get an expanded hem. But in the LHX2 PAX6 double null background, the hem is much bigger. You don't get any anti-hem. Okay, so the hem doesn't form laterally because PAX6 protects that tissue from becoming. I'm going to show you Geeta's work with FOXG1. Geeta showed that LHX2 is a downstream target of FOXG1. Okay, she showed this by examining a ChIP-seq data set in collaboration with Bin Chen, where here you see FOXG1 occupies 3 loci on LHX2, that's the LHX2 gene. And she did a luciferase assay for these specific regions and found indeed that LHX2, FOXG1 binding these 3 loci upregulates the luciferase reporter. And, oh, here's Geeta, sorry, I should have introduced her earlier. She's a research scientist with CIPLA now, and she was a Wellcome DBT early career fellow when she was in my lab. So what have we got so far? Okay, so here in the FOXG1 conditional knockout, which we obtained in collaboration with Goichi Miyoshi and Gord Fischel, by giving low-dose tamoxifen on a Cree-ER background, Geeta was able to induce FOXG1 null patches because she gave low-dose tamoxifen. You can see there is some FOXG1 still expressed in some cells, light purple. And interestingly, where FOXG1 is completely gone, there LHX2 is completely gone. So we did nothing to LHX2. This is only a FOXG1 conditional. Right? But because FOXG1 regulates LHX2, loss of FOXG1 caused complete loss of LHX2 in patches. And where there is weak FOXG1, there was LHX2 expressed. So here too, we have ended up creating a sort of chimera. This time, loss of LHX2 was created by loss of FOXG1, one step upstream. And indeed, where LHX2 is completely missing, only there we got ectopic hems. Okay, so it didn't matter whether FOXG1 was weak and so on. What mattered whether LHX2 was weak or not, what was gone or not. And FP1, again, a hippocampal marker, came up only in the non-hem LHX2 positive patches. Okay, so this then began suggesting that LHX2's job may possibly be beyond just protecting the tissue from becoming hem. Okay, and that's the question I'm going to explore next. But first, let me just summarize. We showed, I didn't show you the PAX6 data, but we showed, it's published, Godbole et al. 2017. We showed that these 3 transcription factors work in concert to suppress the hem. And I showed you here that FOXG1 regulates LHX2. And what we published earlier is that LHX2 actually regulates PAX6. So keeping the hem to a contained location seems to be very important for early development. And this is understandable because where the hem is, there hippocampus forms next door. So keeping the hem precisely positioned is a good thing to do and an important task to do for the embryo. Okay. So the question then is, why does LHX2 expression persist in the hippocampus and cortex long after LHX2's job in hem suppression is over? Once the hem is specified, loss of LHX2 after embryonic 11.5 does not cause transformation of cortex into hem. So does LHX2 have an additional role in the cortex, in the primordium of the hippocampus and the cortex for that matter, beyond protecting them from becoming hem? And I'm going to answer part of this question, the hippocampal part of this question, in the next few slides. And for this, I have to introduce another molecule, not a transcription factor, but a binding partner of LHX2, limb homeodomain binding factor, Ldb1. Okay. So LDBs are cofactors. They bind genes beyond limb homeodomain genes, but here the relevant function is the limb homeodomain binding function. Here is a 10.5 embryo. You can see LHX2 expression. You can see it already excludes the hem. And you can see here Ldb1 with its other names, NL1 and CLIM. Its fly counterpart is CHIP. And you can see Ldb1 is pretty ubiquitous. Okay. It's been well demonstrated in the literature, in papers that I'm going to cite, that limb homeodomain genes bind Ldb1. And here in our hands and our tissue, we show with an IP with LHX2, brings down LHX2 and also brings down Ldb1. So this situation happens in the embryonic brain as well. Okay. Here now is LHX2 expression in the hippocampal primordium, neocortical primordium, and it excludes the hem. And here is LHX2 expression in the Ldb1 mutant. Okay, this is a conditional mutant induced with telencephalon specific EMX1 Cre. And the reason we did conditional is that Ldb1 is important for several other things. So Ldb1 nulls die very, very early. The point of this slide is to show you that loss of Ldb1, the cofactor, doesn't appear to change the expression of LHX2. But as I will show you, it makes it so that loss of Ldb1 prevents LHX2 from actually doing its job. And that's what I'm going to show you in the coming few slides. Okay, so here is a control brain. We're focusing in on the medial wall. This would be where the hem is, and FP1 is a marker for the hippocampal primordium. I'm going to use this marker again and again. You can see how in the cortex-specific loss of both LHX2 and Ldb1, there is very, very little FP1. So the hippocampus is almost completely missing. Okay, is the hem missing? Because I told you the hem was important for the hippocampus. Turns out the hem is actually present, hale and hearty. Okay, but so although the hem is present, and this is Wnt2b, although the hem is present and expressing Wints, the responding tissue is for some reason unable to develop into hippocampus when Ldb1 is gone. The hem is present, LHX2 is present in the responding tissue, but Ldb1 is gone. So that's what I'm going to explore now. Okay, to introduce LDBs. Here in yellow, you can see that the Ldb molecule has two components. Okay, there's a dimerization domain, so it self-dimerizes, and there's a limb interaction domain via which it binds the paired limb domains of LHX genes and a homeodomain which binds DNA. This homeodomain can also complex with other factors or directly bind DNA, but you can see how the dimerization is the key. By Ldb1's dimerization ability, it essentially brings together complete two macromolecular assemblies together. And these macromolecular assemblies can themselves bind DNA. Ldb1 doesn't bind DNA, but because it dimerizes, it can pull together assembly 1, assembly 2 from distant regions in the chromatin and potentially loop the chromatin and bring together interactions between different domains of chromatin, distant enhancers, and so on. Okay, Ldb1 has been known to bind many, many other factors. Today, we're going to focus on its binding LHX2. And we're going to model our analysis on fantastic work done in Drosophila by Milan and Cohen and Van Meyl et al. More than 2 decades ago. Okay, so this big curved loop of chromatin, I've just compressed and I'm representing it here as they do in their papers, a shorter stretch separating them. But the idea is the same, that two molecules of LHX bind the DNA and they are bridged by a dimer of Ldb. Now, they did their work in Drosophila where the LHX2 direct ortholog is Apterus. Okay. Apterus mutants don't have wing defects. And the the Ldb counterpart is CHIP. Now, what they did, because they had a lovely phenotype to score, because they could score whether the wing, so Apterus specifies the dorsal portion of the wing blade. So as you may know, if that's the wing blade, that's the wing disc, okay, there's an oval portion of it which has a dorsal and a ventral component. And as the wing disc elaborates, it forms the fly wing, dorsal and ventral. And what they showed was Apterus null clones in the dorsal component develop ventral characteristics, and overexpressing Apterus in the ventral component develops dorsal characteristics in the wing blade. Okay, so by dint of apterous expression in the dorsal component, the wing blade achieves the dorsal part of its identity. Okay, now in apterous nulls, the dorsal component doesn't develop, so the wing becomes this little shriveled stump. So they had an easy phenotype to score for. And what they did was they rescued this phenotype in a very cool way, not by putting back. So now the apterous phenotype I just showed you, I just demonstrated to you. The chip phenotype is similar because apterous needs chip LDB to work. What they did was they rescued this phenotype by a chimeric construct in which they removed the middleman and they simply had the apterous LHX2 homeodomain bridged with directly the dimerizing domain of chip. And this chimeric construct, they showed, was able to rescue these mutants. Okay, so this basically means that the job of Ldb1 in this context is to bring a dimerization between the two homeodomains of the LHX apterous gene. So a student in my lab, Hari Padmanabhan, created a counterpart of this chimeric construct using LHX2 and Ldb. And that's what we're going to use for the next few experiments. So here is Hari. He has long since left the lab after a postdoc with Jeff Macklis at Harvard. He's now in industry. And this, the experiments I'm talking about are the work of a PhD student, Veena Kinnare, and Geeta Godbole and Archana Iyer did very, very difficult in utero electroporations for this. Okay, so here are the in utros that Geeta and Archana did. Electroporating now the chimeric construct I told you about at embryonic 10.5 and looking 2 days later. And I want to emphasize that we did these electroporations in the loss of LDB background. Okay, remember, loss of LDB allows LHX2 to be expressed, but our hypothesis is that LHX2 can't work. And now we're introducing a construct that is essentially the LHX2 homeodomain with the ability to dimerize. Okay, so here's FP1 in the hippocampal primordium, and here's LHX9. It's in the hippocampal neuroepithelium, and it's also in these cells that line the cortex. So don't look at these. And this is control. This is now the Ldb1 mutant, the non-electroporated hemisphere. You've seen this before. There's barely any FP1 expression. And here, there's barely any LHX9 expression in the hippocampal neuroepithelium, though these cells are present. Now, what happens when the other hemisphere is electroporated with this chimeric construct? Suddenly, you get patches of FP1, which line up nicely with the electroporated green patches. Suddenly you get patches of LHX9 which line up nicely. And here's a lovely HiMag that makes that case. So in the primordium, we're now inducing cells, okay, that cell autonomously appear to have the ability to display hippocampal markers. What happens a little bit later? Do we actually get hippocampus? Well, so Geeta and Archana did electroporations a little later at E1340. And now we're looking at birth P0, okay? The non-electroporated hemisphere has barely a shrunken hippocampus, as I showed you before. And this is now Neuro-D, a transcription factor that identifies the dentate and a little bit of CA3. And look at this sort of streaming line. Here now are 2 more markers. This is ZBTB20, which identifies the entire Ammon's horn of the hippocampus and the dentate. And this is KA1, which is exclusive to field CA3. And look at the non-electroporated Ldb1 mutant hemisphere, hardly any expression of these markers. Now we see the other hemisphere electroporated with Ldb, and you can suddenly see a little patch of Neuro-D come up, and you can see extra patches of both ZBTB and KA1. And here it is in HiMag. Look, we get this dark cluster and a little streaming line, very much parallel to this. And what's interesting, which I want to underscore, is that this marker, which is just dentate and CA3, only comes up in a positionally appropriate way at the very end of this entire tissue. Although there are green cells here that are happily expressing this construct, the field-specific marker doesn't come up in the wrong place. So it's not as if this construct just induces all hippocampal fates everywhere. Those fates are still induced in a regionally appropriate manner, presumably because of graded signals from the HEM, the organizer model I spoke about in the first bit of my talk. Okay, so we have appropriate readout of hippocampal identity enabled by now the presence of this chimeric construct. The same way here, ZBTB, in this oval, I'm outlining the far end of the Ammon's horn, the CA1 end. You can see KA1 doesn't come up there, although there are green cells happily clustered there, but ZBTB does indeed come up there. Okay. Right? Now, I'm going to move on to the next question. Okay. Basically, this whole study so far is telling us that the presence of LHX2 in the hippocampal primordium with the help of LDB, okay, is possibly responsible in helping these cells interpret the Wnt signals and express hippocampal fate. Because although there's LHX2 present in these LDB mutants, it can't act without being dimerized. Introducing this dimerizing component allows now this complex to express hippocampal fate. So, well, does LHX2 help presumptive hippocampal cells to process the Wnt signal? Is LHX2 actually downstream of the Wnt signal? Okay, having worked very hard to suppress hem fate early in the primordium, now it's helping the cells respond to the Wnt fate. Kind of interesting, isn't it? Well, for this, we're doing some bioinformatics studies, okay? And I want to introduce how LHX2 might actually help the cells interpret the Wnt signal by introducing a transcription factor Ldb1, which is expressed in the hippocampus, you can see very nicely, and which is necessary for hippocampal or dentate fate. In the Ldb1 mutant here, Dalsaran and others have shown that the dentate is missing. And in fact, in a slightly dominant negative Ldb1 version, here most of the hippocampus is missing in the same way. So we're going to look at now LHX2 and its occupancy and so on the Ldb1 locus. Okay, so here again, just a cartoon to show you where LHX2 is expressed. And this is the work that was started by Bhavna Muralidhar, who was a postdoc in my lab, a Welcome TBT Early Career Fellow. She's now started her own lab at NCAM in Bangalore. And we are working on wrapping up this last paper first. And this paper was then carried forward by Gautam Suresh, another PhD student in the lab, joined by Mahima Bose, and a PhD student, and colleagues from Sanjeev Galande's lab at IISER Pune, Saurabh Pradhan and Chandramouli Puri. Okay, so what did this team do? Here, they looked at the occupancy and the accessibility and RNA expression in the hippocampal primordium and the neocortical primordium. Okay, I'm only going to show you the data with respect to the Ldb1 locus. So here's the Ldb1 locus. Because the study is not complete, I'm giving you an idea of where we are going. When we look at accessibility, okay, in a taxi, you can immediately see that this Ldb1 locus is more open in the hippocampus than it is in the neocortical primordium. When we look at chromatin activatory marks, either marks near the transcription start site or marks near distal and near enhancers, active enhancer marks, we again see a pattern that corroborates this, that the hippocampal chromatin is more open for this locus than the neocortex. And finally, when we look at LHX2 occupancy, we see interesting differences and unique peaks that are consistent with the idea that LHX2 regulates Ldb1. This is the kind of data that we are examining. The study is not complete yet. We're still processing the RNA-seq results and so on to tie up a complete story. But I wanted to give you a sense of how we are looking at this as a global picture to ask, does LHX2's presence in the hippocampus allow it to become hippocampus? And does LHX2 have different jobs in the neocortex? That's sort of the angle we are taking. And now I'll summarize so far. And in the last 5-10 minutes, I will tell you a cool story that involves human embryonic stem cell organoids. Okay, I've summarized that 3 transcription factors that we know of interact to position the hem, and that an LHX2 Ldb1 partnership is sufficient to specify hippocampal fate, and that LHX2 may regulate the canonical Wnt response within the hippocampal primordium adjacent to the hem to allow it to acquire hippocampal fate. Okay, now we're going to ask a question that's kind of been staring us all in the face for this whole talk, right? This is the hem. It expresses Wnt signals, and I've shown you that the hippocampus is induced. Okay. The key question we want to ask is, why doesn't the hippocampus form on the other side of the hem? How do the Wnt signals only know to go upwards? Kind of funny, right? Well, the answer is they don't. They go in both directions. Okay. And I'm going to now focus on this structure, the choroid plexus, which is produced by the hem. It's a lineage derivative of the hem. And I'm going to show you that it needs a fine balance of Wnt signaling. Okay, if we have too much or too little canonical Wnt signaling, the choroid plexus is affected. All right, for lack of time, I'm going to compress this portion of the talk just to give you an idea of where we are at. So here I show you that the choroid plexus in the embryo expresses components that are consistent with it processing canonical wind signaling. Beta-catenin, a critical fat part of the canonical wind pathway, and Lef1, and here frizzled, a receptor, AXIN2, a readout of canonical wind signaling, TTR, a marker for bona fide choroid plexus. Okay, and here is a reagent I'm going to use for the last bit of my talk. It's LMX1A Cre. This is LMX1A expression. It's in the hem and the choroid plexus. And when this LMX1A Cre animal, which we obtained from Kathy Millen, is crossed with an AI9 reporter mouse, stop flocks reporter, you can see it faithfully recapitulates the hem and the choroid plexus expression domains of LMX1A, and also the Cahal-Retzis cells, which are a lineage derivative of the hem. So everybody's read that should be read. Okay. Now, in collaboration with Natasha Jovanov-Milosevic in Croatia, we examined whether the choroid plexus in the fetal human also processes canonical wind signaling. And the answer is indeed it does. These are low mags, these are high mags. You can see frizzled 1, beta-catenin, both canonical wind components expressed. And here's aquaporin that identifies the choroid plexus. Okay, so now I'm going to test whether perturbations of canonical wind signaling affect choroid plexus in the mouse. Okay, and I'm not going to show you how we remove the data consists with loss of function of beta-catenin, but basically it's a floxed mouse that removes 1 through 6 exons, and I'm not going to show you this data. Just tell you that the choroid plexus is disrupted. But gain of wind signaling in the choroid plexus is what's interesting. Gain of wind signaling, we used a mouse in which exon 3 is floxed. And when exon 3, which contains the phosphorylation site where GSK3 beta would phosphorylate exon 3 and tag it for degradation, when this exon 3 is removed, beta-catenin can no longer be tagged for degradation. So it remains constitutively active, and that's how we get a gain of function of beta-catenin. And here is what happens when the choroid plexus, which normally does process and respond to wind signals, is now forced to do them in an amped up, overactive manner. Okay, this is the work that was initiated by Mallika Chatterjee, a Wellcome DVT early career fellow in the lab. She now has her own lab at Amity University in Noida, and 2 PhD students, Arpan Paricha and Gautam Suresh. Okay, so what does beta-catenin gain of function do in the choroid plexus? The short answer is that too much canonical wind signaling suppresses choroid plexus fate. It begins to not express markers of choroid plexus, TTR, for example. Here's AXIN2 showing whopping amounts of wind signaling, which are normally very weak. And now it begins to express neuronal or hippocampal markers. PROX1, which is normally never expressed in the choroid plexus, nor NeurOG2, are now both expressed in this gain of function choroid plexus. This is by in situ. We can also show it by doing transcriptomics. So here's how we dissected the choroid plexus. And Varun did this nice PCA analysis, which shows you that the mutant choroid plexus hangs out closer to the control and hippocampi than near the control choroid plexus. And here is a heat map showing again control choroid plexus, gain of function choroid plexus, which is looking more and more like the control hippocampus. Look at the similarities between the genes. Okay, choroid enriched genes are down in blue and hippocampal enriched genes are up. Okay, and here again is just detail on this. What you can see is that in the gain of function, choroid enriched genes, aquaporins, channels, transporters are all down. Okay, so the choroid is no longer the choroid. It neither can secrete fluid, so the ventricles are collapsed, and nor does it have a range of transporters that are necessary for choroid plexus function. Okay, just to remind you. Okay, well, we explored whether this transformation or suppression of choroid fate is paralleled in the human. And for this, Arpan traveled to the Weizmann Institute to Orly Reiner's lab, where he actually learned to make human embryonic stem cell derived organoids. Okay. Here's Arpan. And here is the organoid protocol, starting with embryonic stem cells. They are cultured for 18 days, at which point they become these lucky little balls. And then a standard morphogen cocktail is added. Okay, this is a protocol established by the Sasai lab that normally induces choroid plexus fate in organoids. And what does this cocktail involve? This cocktail involves some BMP signaling and some activation of Wnt signaling by this drug here. Okay, this is an activator of Wnt signaling in that it works similar. So basically the effect is similar to the beta-catenin gain of function. It essentially stabilizes beta-catenin. Okay. And in Arpan's hands, these organoids develop. So up to 18, they're rounded. Then they begin to develop these blebs. These blebs at their periphery express choroid plexus marker TTR. So the organoid protocol worked. It expresses choroid marker OTX2. And in fact, looks very, very much like the mouse section. So this is aquaporin, which is distributed only on one side of the mouse choroid plexus, and this is our organoid. So it begins to look pretty similar. I'm going to show you what happens when these organoids are treated with increasing concentrations of the Wnt, canonical Wnt activator KIR. This is day 25, and everybody is happy expressing choroid markers. The minute we put more KIR, all of these choroid patches disappear. Okay, and here is the quantitation. TTR, which had nicely gone up by day 18, disappears. Aquaporin disappears. OTX2 disappears. But LEF1, which is a canonical Wnt factor, increases as expected. Okay. Here, a few more examples and a few more markers. And here's a range of the KIR treatment. You can see TTR disappears, aquaporin disappears, OTX2 disappears. But hippocampal marker ZBTB20 appears to come up. Okay. Now, we looked at this with just one marker. We don't have a range of hippocampal markers to claim that we have induced hippocampus in these cells. But indeed, we have suppressed choroid plexus fate in these cells. Okay, we've done quantitations and we've done RNA-seq to conclude quite well that the choroid plexus fate does indeed get suppressed. So here, from day 18 to day 30, choroid has been induced. And then comparing 3 micromolar KIR with 12 micromolar, all of those genes basically get tamped down. So aspects of what we saw in the mouse, we have recapitulated in the human embryonic stem cell organoids. So I'm going to summarize. What did we see with the organoids? I didn't show you the loss of function of beta-catenin, but that the choroid plexus gets disrupted. In the gain of function, we lose the choroid fate brown and we appear to gain neuronal fate in the mouse. And in the organoids, we have shown conclusively that we lose choroid fate. And with one hippocampal marker seems to get induced. But that's not conclusive of hippocampal fate. Okay. I want to wrap up with asking, how does the hem fit into a broader picture of telencephalic patterning? Okay, because this is hem, it induces the hippocampus. But do you have these random organizers here and there, or is there any structure to it? And for this, I want to introduce Aishwarya Raj, who has just started her lab at JNCSR in Bangalore. It's delightful. It's fantastic when one's trainees branch out on their own. And I want to introduce how Aishwarya looked at structures beyond the hem. Remember, loss of LHX2 causes expansion of the hem? Well, Aishwarya showed that it causes an expansion of 2 other key structures. Okay, this is the telencephalon, and this flap here is actually the diencephalon, half the diencephalon. At the junction of the telen and dien is the structure called the thalamic eminence in blue. It's sort of like the thalamic hem, if you will. Okay, but it's called the eminentia thalami. And up front, okay, the hem curves around the way the hippocampus does, but up front, there's no hem, there is in fact septum. So in fact, the reason the hippocampus is a caudal structure is that the hem is a caudal structure. Okay, in the front, you have septum, and that flows contiguously with the hem. Well, Aishwarya showed that while loss of LHX2, we know...
Shubhatole, I'm sorry, I have to interrupt the last 2 minutes. Is that okay?
Sure. Yeah.
Thank you. Thank you.
Maybe 2 slides left.
Yeah.
So what Aishwarya showed is that both the septum and the thalamic eminence also expand upon loss of LHX2. Here is the septum expanding, okay, and here is the thalamic eminence expanding. And what does this mean? This means that LHX2's job seems to be more than just protecting the hippocampus and cortex from becoming hem and anti-hem. It's in fact protecting the cortex from becoming anything other than cortex. Because when you lose LHX2, you get all of these other surrounding fates creeping in and taking over the space. Right? So our collaborator, Gundela Mayer and Alessandra Pirani, with whom we collaborated on this project, Gundela named this the Forebrain hem system. Okay, and she found that the forebrain hem system is in fact paralleled in the human brain as well. So this is the mouse section, and this is from Gundela's collection, a human section. And all these structures exist in the human brain too. The thalamic eminence, the hem, the septum, and so on. This was known. So I'm just showing it to you in parallel, and I'm going to conclude with an evolutionary summary. Okay, the hem and the septum and the thalamic eminence are present in mammals and in birds and in reptiles. But the hem has not been reported in fish and frog. So this suggests a sort of evolution of this inductive system, okay, that creates specialized fates in the cortex, and of which the hem seems to be the most recent entrant, all of which are controlled by LHX2, in that they are suppressed by LHX2, and only when LHX2 is not there can these fates develop at the peripheries of the cortical primordial. So thank you very much. My summary then, how to make a hippocampus. You make, an organizer and a responding tissue. You restrict the signaling center to the correct location. You regulate responsiveness in the adjacent tissue, the Ldb LHX2 complex, for example, and regulated response to wind. And then you nest your organizer in an evolutionarily conserved system so that the brain can evolve. All right, thank you very much. I thank my funding agencies and those that funded the Welcome DBT Early Career Fellows. Veena Kinnare was funded by the UGC, and we have intramural funding from the DAE, which we acknowledge. The organoid work was supported by a Canada-Israel Health Science Initiative grant, in which I'm a collaborator. Thank you very much. I don't know if there's time for questions, but I'm happy to take some.
We do have. Thank you so much, Professor Shubha Tole. So this was really mesmerizing, and your new story of the organoid is very interesting. And unfortunately, for the want of time, I had to intervene. I think we'd love to have you summon to our campuses and listen more. Yeah. So I think we'll take a couple of questions. I don't want to disappoint folks who have posted. So Shruti Shri asks if wind signaling is responsible for hippocampal neuron formation in adult mouse also, because we know the hippocampal neurons can.
Very interesting question. I don't know. I don't know also how to do this experiment, because you would have to have a Cree driver that removes wind signaling only in the adult. The drivers we know of are active from development. One would have to have an engineered mouse, so that's why it's hard to ask that question. But maybe somebody can do it in culture.
Yeah.
Was the hippocampus-specific brain organoid co-cultured with other cells? No, we didn't co-culture with other cells. We induced embryonic stem cells to become, to express choroid plexus, actually. And no co-culture was done. I think the longest we kept them was 40 to 50 days. Mice with many hippocampi, do they survive to adulthood? I already pre-answered that during the talk, that they die embryonically because they don't form erythrocytes in the liver. Is it possible to retrieve damaged hippocampal neurons by stimulating Wnt signaling? So remember, Wnt specifies hippocampal fate. That means it works on proliferating progenitors that are going to become produce hippocampal neurons. Once the cell is a hippocampal neuron, it's not necessary that Wnt signaling will be required to make it so. So again, I don't know. What happens to LHX signaling when the brain is under inflammatory threat? Have no idea. Would you like to join my lab to find out? You guys are all taking me to the edge of I don't know. Cool. Meenakshi says, wonderful talk. Thank you. I understand there's a correct position of the organizer. Is there a time window when these processes take place? The organizer is positioned early. So we did all our experiments where we induced with TAM at 9.5. After 10.5, you can't move the organizer around. So the organizer position acts early. The rescue experiment was carried at E13-14. This is the Ldb1 chimeric molecule rescue. Good catch. We ourselves are very intrigued by that. Now, the rescue experiment didn't do anything to the organizer. The organizer was already there. The M was there. What did we do in Ldb1 mutant responding tissue? We put in now the chimeric molecule. So remember, E13, 14, there was still plenty of progenitors. So we introduced the chimeric molecule into the progenitors when apparently they are still able to suddenly now say, okay, now we can respond to this Wnt signaling and become hippocampal neurons. Right? So presumably because we still had progenitors and this function seems to work in progenitors, we could do the rescue at that early age, but it's pretty intriguing that it did work at that somewhat late age, and very, very good catch. Very nice catch. Okay, can I keep going?
Yeah, maybe we can take one more question. There's one last there. Yeah.
Okay.
Shubhra.
Which other parts of the brain can take place or neocortex when LHX2 is absent in the limbic system? So very interestingly, Shenju Chu, when she was a postdoc in Dennis O'Leary's lab, showed that when LHX2 is lost at 10.5 versus 11.5, okay, you get an expansion of the olfactory cortex, paleocortex at 10.5, but not at 11.5. Okay, she has a Nature Neuroscience paper, 2009, I think you should look, it's a lovely paper. Okay, and Shenju and I wrote a review that summarizes all things LHX2. LHX2 also has cell-fate specifying roles in the neocortex. So if you just look at Chu and Tole, you might get the whole thing. Or maybe I'll put that review in the chat box.
All right.
What about some variants expressing less amount? Dose requirements of LHX2 we haven't looked at. We've looked at all on or off. We haven't looked at graded levels of LHX2. So very, very nice questions. Wonderful questions. I'm delighted to interact with this kind of an audience. I so wish it could be in person, but okay.
Yeah, I think we'll have this session somewhere soon in person. And so thank you so much, Professor Shubha Tole, for this exciting talk and a very communicative, interactive answering session. That was wonderful. So with this, we'll take a short break and we'll come back at 17:53. I mean, we'll reduce the break to short time and then we'll assemble for the lightning talks. 16:53, I'm sorry, 16:53. I also would like to welcome few members who have joined us. Professor B. Vengamma, who's the Vice Chancellor cum Director for SV Institute of Medical Sciences, Tirupati. She's a neurologist, senior neurologist in the city. She has joined us. Welcome, Professor Vengamma. I'd also like to thank Senior Professor M.K. Matthew from NCBS, who has joined us today. I'll also like to thank all the speakers. Speakers of tomorrow have also joined us to listen to the speakers today, and that's exciting. And the judges have joined us. We'll introduce them in a while. And thank you to all the ICER TVM and ICER TPT faculty who have joined us online right now. Thank you so much. With that, we'll meet at 16:54. Yeah.
So I just put a bunch of papers in the chat if people are interested in reading them.
Yeah, thank you. Thank you.
Hello, everyone. Welcome back. And my name is Harshini Chakravarti. I'm from IISER Tirupati. And now we're going to have the next part of the symposium, which is the lightning talks. These are short 5-minute talks by PhD and postdoc candidates. And these are actually meant to mimic the poster session that we would normally have in a symposium like this. So we have some exciting prizes also for the best talks. And we welcome our judges, Dr. Nivedita from NBRC, Dr. James Clement from JNCASR, and Dr. Naren Kumar from IISc. So I'll just give you some instructions to the speakers. The talks are strictly for 5 minutes. Okay, you will hear one warning buzzer at 4 minutes and another one at 5 minutes. And we are not going to have a live question and answer since the time is very short, but the audience is welcome to ask questions in the chat box, and we request the speakers to answer the audience questions in the chat box itself. Okay, so let's start now. The first talk will be given by Aishwarya Raj, who is a PhD student from NIMHANS, Bengaluru. Aishwarya, can you share your screen?
Yeah, sure. Good evening, everyone. Just give me a second, then I'll just share. Could you tell me if it is visible?
Yes, it's visible. You can start.
Okay, it's full screen, right?
Yes.
Okay. So today I would be talking about the deleterious effects of extracellular alpha-synuclein on astrocytic function. This is basically with context to the Parkinson's disease. So I wouldn't be going much into the introduction for Parkinson's disease because we all know it's a neurodegenerative disease with characteristic loss of DA neurons in the midbrain region, and with the occurrence of signature alpha-synuclein and ubiquitin contained Levi bodies. Now, this alpha-synuclein aggregation is not just limited to DA neurons. It is already observed in the CSF or in dystrophic astrocytes, which was checked after the postmortem of PD brains. So most of these studies establish that alpha-synuclein is available in the CNS microenvironment during disease progression, and so it might be likely that the midbrain niche cells are getting affected first before the neurodegeneration. So we have evaluated the effect of midbrain astrocytes for their function when treated with alpha-synuclein. Now, astrocytes are generally in non-reactive states, broad and flat, which I have shown in an immunofluorescence data, but this is tagged with GFAP actually. But after treating them with alpha-synuclein, we are seeing that there is thinning of the processes. The astrocytes become long, and alpha-synuclein is the green one, so it is getting it is interacting with astrocytes, and there is membrane binding in some cases, there is internalization in other cases. So we have treated with wild type monomer and aggregate and double mutant A30P, A53T double mutant monomers and aggregates, and we are finding all of them are interacting with astrocytes. Now, there are a lot of roles of astrocytes that is, very important for neuron survival and defense or differentiation and neuroprotection. Out of these, I have looked into two things. One is oxidative stress prevention, and another is glutamate excitotoxicity prevention. Now, what we did is we have already seen there is increased oxidative stress in these alpha-synuclein treated cells. So the first marker that we would look into is what is the glutathione content and the expression of related enzymes in these cases, because astrocytes are one of the major sources for glutathione in these in the brain. So what we found is in the treated astrocytes compared to the control, the glutathione concentration reduces very much, especially in the mutant aggregate treated ones. The next thing we checked is how is the glutathione synthetase enzyme, the gene expression of it, and that has also come down in a similar trend to the glutathione concentration. Further, we looked how is the glutathione reductase and the glutathione peroxidase enzymes, which are also very important to curb or to neutralize the oxidative stress, and both have again reduced immensely. And the other function that we looked into is glutamate content and how it prevents excitotoxicity. Now, what happens here is, yes, there is an increase of glutamate content in these cells. So, there might be more pickup of glutamate from the neighboring niche area, which was represented by the Gl1 flow cytometry that we did. Now, Gl1 is one of the major transporters for picking up glutamate from the CNS microenvironment to the astrocytes. So, yes, there is excitotoxicity prevention, but what is happening to the glutamate inside the cell? Is it being converted to glutamine because the next neuron requires glutamate in the form of glutamine only? Only glutamine can be transferred to the neurons for, the glutamate-glutamine cycle is like that. So is it being converted to glutamine? And that with this glutamine synthase gene expression, we found that that is pretty much low in the case of the alpha-synuclein treated cells. So that means that there is accumulation of glutamate happening inside the cells. Now, what happens if there is accumulation of glutamate? That is the next question. Apart from that, we also did pyruvate carboxylase gene expression. What this enzyme does is it helps in the de novo synthesis of glutamate in the cells. Now that is also increased in these astrocytes. So, sorry? Hello?
I think your 5 minutes are up.
Okay. Yeah. So yeah, that is how what I would like to say, that there is a dysfunction of astrocytes. Plus, we might need to consider these niche cells for therapeutic strategies. That's all. Thank you.
Thank you, Aishwarya. It was a lovely talk. Next speaker we have is Gautam Suresh, PhD student at IISER Tirupati. Gautam, you can share your screen.
Hello.
Hi. You need to share your screen.
Yeah, I hope everyone can see my screen now.
Yes.
Yeah. So, hello, good evening, everyone. Today I will discuss about how metabolic stress affects the neurites of adult-born neurons. The establishment of precise connections in the nervous system throughout the development is important for proper functioning of the brain. The neurite outgrowth process is the initial step towards making connections between the neurons. As soon as the neurons develop, they send out budding neurites and they elongate to form axon and the dendrites. So the growth cone is the driving wheel for the neurite to form the synapse. Compared to the neurites in the developing brain, which is highly proliferative, in the adult brain, the neurites maintain synaptic plasticity and are more susceptible to stress, which can lead to neurodegeneration. The extension of axonal and dendritic process in the CNS is tightly regulated by outgrowth promoting and inhibitory cues to assure precision of synaptic connections. First, let me explain briefly about the molecular factors. There are several factors such as Caspar, prion, NCAM, laminin, and CEPP alpha. Out of this, cell adhesion molecules play a critical role in determining the neurite length and branching. For example, here you can see the Caspar knockout, which leads to the higher amount of increase in neurite length compared to the wild type. So here the Caspar one is an inhibitory protein, whereas another cell adhesion protein called the prion is a neurite outgrowth promoting molecule. Here I will show you a short video how it works. So here you can see the growth cone moving towards the recombinant PRP that is being added to the device, like microfluidic device in the in vitro condition. Here you can see the growth hormone moving towards the recombinant ER3 protein. So here it is a promoting, a neurate outgrowth promoting protein. Next, the metabolic factors. Reduced supply of oxygen leads to hypoxic condition in the brain. Oxygen deprivation activates Hip1 alpha, which in turn affects the physiology of the brain. So this will lead to this activates the Hip1 alpha, which affects the VASP and also leads to the cytoarchitectural collapse. As you can see in the graph, the hypoxia condition leads to reduced neurate branches compared to the normoxic condition. Next metabolic factor I'll talk about is the glycemic stress. Here you can see the hyperglycemia will induce the impaired insulin signaling, which will activate the apoptotic signals and leads to the death of the neuronal cell. Here you can see in the hyperglycemic condition, the neuronal projections are lost and the neuronal extensions is like highly affected over here. So what is the research question we address? How the oxygen and the glycemic stress affects the downstream signaling and metabolic pathways in the adult neuronal cells, which can lead to neurodegeneration. To address the research question, we established the adult neuronal culture, then induced metabolic stress by culturing the neurons in the hyperglycemia and hypoxic condition. Then we do the assessment of cell viability, gene and protein expression, neurate morphology, and real-time growth hormone dynamics and mitochondrial metabolism. So earlier we have checked the effects of hyperglycemia in the adult retinal neurons, so which enhance the neurate length and also down-regulating the cell addition molecules. Here I haven't shown the data of the cell addition molecule, whereas we can see the number of cell addition molecules that is down-regulated in the hyperglycemia and also promoting the or enhancing the neurate length, which is not the normal physiological condition of the neuron. And this was published in Frontiers in Neuroscience in 2019. Currently, we have established a hypoxia system where we culture the neuronal cells and in a hypoxic condition, that is 2% oxygen, instead of normoxia, which is 20% oxygen. And we have observed interesting morphological changes in the neurates compared to the normoxia condition. So the future directions of our project are to check the effects of metabolic stress in the cerebellar, cortical, hippocampal neuron, and also in the retinal explants. The in vivo experiments have to be done for the better understanding of neurodegeneration caused by this metabolic stress. And the significance of our project is long-term exposure to hyperglycemia in diabetes, which leads to the neurodegeneration. And also to find out the critical roles of cell addition molecule in regulating the neurodegenerative process in controlling or enhancing the neurodegeneration. Thank you. I can take questions if there are any.
Thank you, Gautam. But there's no questions here. So let's move on to the next speaker, who's we have with us Niyama, who's a PhD student from IICT Mumbai. Please share your screen. Yeah, you can see it.
My screen is visible to all, and am I clearly audible?
Yes.
Just a second. So good evening and pleasant greetings to all. I'm Niyama Chandhanawala from Institute of Chemical Technology, Mumbai. The title of my research work is aposinin, which is a nootropic, neurotrophic, and neuroprotective treatment modality for Alzheimer's disease. So what is Alzheimer's? It is an irreversible progressive brain disorder that slowly destroys memory and thinking skills and eventually the ability to carry out the simplest of tasks. The age-specific prevalence of AD doubles every 5 years post the age of 65. And there are over 9.9 million new cases of dementia each year, implying 1 every 3.2 seconds. The average cost of treatment is nearly $215,000, which is a lot. The currently available treatment for Alzheimer's, there's a new approved drug in 2021, which is aducanumab, which targets mild cognitive impairment individuals only in the early stages of dementia. The classic hallmarks of Alzheimer's are neurofibrillary tangles, which are present intracellularly, and extracellularly amyloid plaques are present. The top 10 early signs of Alzheimer's include changes in vision, poor judgment, memory loss, among others. And the pathological features include immense oxidative stress, mitochondrial dysfunction, altered oxidative phosphorylation system, inflammation, among others. In light of this, we chose to study aposinin, which is an aromatic ketone obtained from Precoriza curoa roots. It is an NADPH oxidase inhibitor, which reduces the production of superoxide anions. It also has a multidimensional therapeutic role, not only in neurodegeneration, but also in conditions like atherosclerosis, hypertension, and stroke. We hypothesize that apocynon will reduce the reactive oxygen species generation, thereby preventing neurodegeneration. For this, we investigated the neuroprotective potential of apocynon and an Alzheimer's disease sporadic rat model employing amyloid beta introduced intrahippocampi and studying the physiological alterations. After injection of amyloid beta, after a recovery period of 1 week, we studied on the 1st and the 20th day with open field test and Barnes-Maze test for exploratory behavior as well as spatial learning memory, after which animals were sacrificed and then the biochemical parameters were assessed. In the neurobehavioral activity, it was observed that at doses of 150 and 300 mg per kg, apocynon significantly decreased the grooming behavior. When we check for spatial reference memory learning, we observed that at 50 mg per kg dose, apocynon significantly reduced the escape latency. Then we went on to study the antioxidant and anti-inflammatory behavior, wherein the natural defense markers, SOD, GSH, and catalase, they showed a significant improvement, whereas the levels of LPO, we showed that apocynon at all the 3 doses significantly reduced the levels of malondialdehyde. And nextly, we saw in TNF-alpha, which is a pro-inflammatory cytokine, apocynon was showing a prominent effect. Next, we studied the anti-apoptotic activity for apocynon, wherein it was observed that the caspase-3 expression was significantly reduced and effects can be seen for all the 3 doses. When we performed concurrent staining for amyloid beta, the intensity was significantly enhanced. And although it was not significant, apocynon did reduce this certain intensity. We also performed different staining techniques wherein we showed that amyloid beta deposits were reduced with the help of apocynon, and a prominent effect was observed at 50 mg per kg. Next, we went on to explore the adult hippocampal neurogenesis activity, wherein we saw that at for levels of BDNF, Ki67, as well as DCX double cortin, we saw that apocynon was showing a prominent increase in the neuronal activity. The intensity of these Ki67, DCX, as well as BDNF neurons was increased. Lastly, we used PGC1 alpha, which was a marker, a master regulator of mitochondrial biogenesis, and we showed that apocynon at dose of 50 mg per kg significantly enhanced the expression. Hence, I'd like to summarize that apocynon not only gave us a nootropic effect, but also antioxidant, anti-inflammatory, decreased number of amyloid deposits, anti-apoptotic, neuroprotective, neurogenesis, as well as mitochondrial biogenesis. With this, I would like to sum up my presentation with just one question. Is a world without dementia a possibility? Thank you very much for your patient listening.
Thank you. Very interesting. Next speaker we have is Srita Sharma, who is a PhD student at the Department of Physiology, AIIMS, New Delhi. Hi, you can
Good
share
evening.
your screen.
Yeah.
Good evening. Yes, your screen is visible.
Are the slides moving?
Yes.
Shall I start now?
Yeah.
Good evening to all present here. Myself, Shweta. I am second year PhD student in Department of Physiology, AIIMS, New Delhi. My PhD work is on effect of probiotics and repetitive transcranial magnetic stimulation on gut-brain axis in 6-hydroxydopamine rat model of Parkinson disease. Parkinson disease, as we all know, that is second most common neurodegenerative disease, which leads to progressive deterioration of motor functions due to loss of dopamine producing neurons in substantia nigra, which is clearly visible in this brain sections as well. The lighter color is due to absence of dopamine in substantia nigra. Many studies have been hypothesized that there is gut-brain axis present in organisms, and any dysfunctioning in the gut leads to dysfunction in the brain. And nowadays, the consumption of various environmental toxins in the form of pesticides, organic solvents, heavy metals enhanced, due to which gut microbiota disrupts. And the disruption of this gut microbiota leads to disrupted epithelial barrier, due to which many toxins enters the brain through the axis, and this leads to Parkinson's disease. The current therapeutic strategies are the consumption of levodopa, but the chronic use of levodopa possess many side effects, including non-motor symptoms, including bowel syndromes, constipation, etc. So it should be replaced with newer therapeutic modalities, including these various. And in our lab, we are using magnetic field stimulation for the brain axis and probiotic stimulation for the gut axis. And various studies have been done on probiotics individually, which shows that probiotics modulate the gut and improves all the deficits of PD via producing short chain fatty acid, which strengthen the intestinal epithelium, also improve the selectivity barrier of intestine, and enhance the immune system. Similarly, the RTMS also, by stimulating our motor cortex, enhance the expression of various factors, including BDNF, GDNF, and the TH positive neurons, and also improves deficits in behavior. But there is no study with RTMS on blood-brain barrier, gut permeability, and beneficial gut microbiota. And so in my study, I retrieved the lacunae that RTMS, there is no study with RTMS on gut-brain axis, and the combined effect of probiotics and RTMS have not been studied as neuroprotective benefits. So I hypothesize that administration of these probiotics along with RTMS may improve the neurological functions via gut-brain axis in PD rats. So in my study, the work design is on first day 6-hydroxydopamine is induced, and after 7 days, these intervention individually as well as in combination administered to the rat for 4 weeks. And at the end of 4th week, various behavior tests again performed and sacrifice the rat. And these parameters like patch clamp technique, patch clamp for striatal neurons, and IHC on various factors for neuronal survival TH positive neurons, BDNF and GDNF will be analyzed on brain as well as colon sections. And morphology will be checked with the help of staining, that is H&E. And gut microbiota assay will be done with 16S ribosomal RNA sequencing technique. Why I choose 6-hydroxydopamine? Because it is specific for the dopaminergic neurons. Secondly, it progressively degenerate the dopaminergic neurons, not spontaneously. So it mimics human PD. And the main mechanism is it causes oxidative stress in the neurons, which leads to Parkinson's disease. So my area of interest in brain is striatum. By using Pexinose and Watson brain axis, striatum neurotoxin, that is 6-hydroxydopamine, is injected in the striatum. And for colon, the area of interest is the ganglion cells. Anticipated results that after the intervention, various deficits in the behavior may improve. The morphology also improves, and the expression of TH positive neurons both in colon and brain improves, and there will be enhancement in the expression of BDNF and GDNF. I would like to thank my guide and co-guide, and thank you.
Thank you. The next speaker is Aishwarya Ramakrishnan, PhD student from JNCASR. Aishwarya, are you there?
Yeah. Can I start sharing?
Yeah.
Okay. Can you see this?
Yes, you can start.
Okay. Yeah. Good evening. I'll be talking about my PhD work, which is looking at the role of electrical synapses, which is something different from what people have been talking about here, and their role in modulating a very core property of circadian clocks using Drosophila melanogaster. So what are circadian clocks? They are internal timekeeping mechanisms which restrict your behavioral and physiological activities to certain times of the day on a 24-hour scale, as you can see here. Okay. So the main function of a circadian clock is to synchronize your activities to the external cycle, for example, light and dark, which happens on a 24-hour scale. But even if you don't have such external cues, your clock is going to keep running with an endogenous period, which is called the free running period, and which is approximately 24 hours. So we study circadian clocks using Drosophila melanogaster, and the circadian clocks in these flies are about 50 neurons which are distributed bilaterally in the brain, and they are responsible for orchestrating several rhythmic behaviors. Now, each of these neurons have a molecular clock which is characterized by the presence of a transcription translation feedback loop, and this entire loop takes about 24 hours to complete, and this is self-sustaining in the absence of any external cue. Thus, a wild-type fly will have a free-running period of about 24 hours. The rhythmic oscillation of these clock proteins in these neurons, it mirrors the rhythms in activity and rest. So we know from the current literature, we know that although each of these neurons have a molecular clock, for proper functioning of the circuit, the neurons in the circuit, all the 150 neurons, they need to extensively remain connected and communicate among each other. And this is very important for generating coherent behavioral rhythms. Now we know that, as you can see in the picture over the right, that there are extensive communication via chemical synapses, which uses neurotransmitters and neuropeptides. What I asked was, is there any role for the other kind of synapse, that is the electrical synapse, which is basically made up of gap junctions, and they are direct cell-to-cell connections. And these gap junctions, as you can see in the picture over the right, down, these are made up of proteins called as connexins in vertebrates and innexins in invertebrates. So to ask if innexins play any role in the clock network of Drosophila, we started with a knockdown screen where we downregulated each of the innexin genes, which are 8 in Drosophila, individually using a Galfo driver, which is called the TIM Galfo driver, which is going to target all these 150 neurons. And as you can see in the graph, we see that there is a lengthening of free running period. Now, instead of a 24-hour wild type period, when you knock down innexin 1 and innexin 2, you get a lengthening of free running period, which is about 1.5 hours more. Like they run with a period of 25.5 hours. So we concluded that knockdown of innexin 1 and 2 lengthens the free running period of activity rest rhythms, which means they are necessary for maintaining the normal 24-hour period. We also specifically mutated the gap junction forming domains of innexin 1 and 2. And we found that when you mutate only the gap junction forming domain, you still see a lengthening of free running period, which is that these proteins are actually forming gap junctions in these neurons, and they are acting as electrical synapses, and an absence of this will lead to a lengthening of free running period. So we put an antibody against the innexins, as you can see in the red channel here, and we see that they co-localize with about 8 neurons, which are the central pacemaker neurons in this circuit. And they are also present in the optic lobes, which are basically giving the light input to the clock. So now we specifically downregulated these innexins, innexin 1 and innexin 2, using a driver which is going to target only these specific 8 neurons on both sides of the brain. And we again see that there is a lengthening of free running period, which tells me that these proteins are present in these subset of neurons. We looked at the oscillation of the core molecular clock in these neurons by staining for a core molecular clock protein period over a 24-hour cycle under constant conditions. And the gray graph that you see, it shows that there is a 24-hour oscillation, that is a peak and a trough in the protein in wild type. Whereas in red, which is innexin 1 knockdown, you see that now this clock is shifted. It's shifted towards the right, which means there is a delay in the accumulation of this protein. Similarly, for knockdown of innexin 2, again, you see that there is a shift in the molecular clock. So we conclude that knockdown of innexins 1 and 2 delay the molecular oscillations, and this suggests that these gap junction proteins are acting via the molecular clock to regulate the free running period. So next, we looked at the release of a neurotransmitter by these neurons by looking at the axonal projections of these neurons. This neurotransmitter PDF, or pigment dispersing factor, is a very important circadian neuropeptide.
Aishwarya, your time is up.
Yeah, I'm done. Which is necessary for orchestrating behavioral rhythms. And we find that in the absence of innexins, the level of this neuropeptide is much higher in the brain, which tells me that probably the release of the neuropeptide is affected. So we summarize that these two proteins, innexin 1 and 2, are necessary in these neurons to for a proper molecular clock oscillation, and an absence of this probably via affecting the membrane is going to affect the neuropeptide release in these neurons. And these are some of the future experiments and acknowledgements. Thank you so much.
Okay, thank you. Next. The next speaker is Hriday Pandey, a PhD student from NBRC. Hriday, can you share your screen? Yeah, we can see your screen. But we can't hear you.
So can you hear me?
Yes, now we can hear. Yeah.
Okay, should I start?
Yes, you can start.
So I'm going to talk about the protein molecule called as corrin 1A, which facilitates calcium mobilization in human astrocytes. So first of all, so what happens in the case of astrocyte activity and reactivity is that this molecule calcium plays a central and a key role in mediating the release of growth factors, gliotransmitters, cytokines, chemokines, and many other factors which we really don't know. So while looking at the regulators of calcium, one protein grabbed our attention, which is called as corrin 1A. And while searching for its role, I found out that it regulates T cell activity via calcium, and it also regulates their differentiation, their survival, their activity, T cell receptor mediated activity. So I thought it might play a role in the activity as well as reactivity of astrocytes also, because calcium is again it has been found to be playing very important roles. So to carry out our experiments, what we do is we derive neural progenitor cells from the aborted fetal samples, which we collect from the nearby hospitals. And from the area called as lateral ventricles, we derive these neural progenitor cells. We can differentiate them into astrocytes and neurons, which takes 3 weeks by giving them particular and specific media conditions. So once we are sure that we have our mature astrocytes and neurons ready with us, we start our experiment. And first we wanted to check the expression of corrin 1A. So here is the Western blot, ICC and IHC images results, which show the expression of corrin 1A in astrocytes. And the expression of astrocytes was validated in human brain autopsy sections as well. So after checking the expression, we went ahead and we wanted to know the role of this protein in resting state conditions. So nothing was known about it. So we started with a loss of function approach. We knocked it down using siRNA. As you can see, the transcript and the protein levels of this protein get downregulated. And we went ahead to check the survival as well as the proliferation of these cells, but nothing was found to be affected. Also, the main proteins, which are intermediate filament proteins called GFAP and vimentin, which are also considered as a cell activity marker, these were also found to be unaffected, unaltered at all. So next, we went ahead. So it was a main finding that the resting state physiology of these cells is not affected at all by these cells. So we went ahead and just a second. Yeah. So next, I wanted to check whether the roles of this protein are activity dependent. So I wanted to stimulate these astrocytes, and we used ATP to stimulate them. And I found that the ATP-induced calcium mobilization in these cells was significantly impaired when we downregulate corrin 1A using siRNA. So this was a very important finding, because calcium plays a very important role in activity and reactivity both of these cells. So if a molecule is facilitating calcium mobilization, so it might also regulate the calcium-mediated events in these cells, which are also a part of the reactive status of these cells. So next, what we did was, we also found that, we turned these cells reactive using HIV1 TAT expression vector. So HIV1 TAT expression vector, turns these cells reactive, which is characterized by increased GFAP concentration, GFAP expression, IL-6 release, calcium mobilization is there. And there is augmentation of various pro-inflammatory genes and proteins as well. But HIV1 TAT, which has been reported to turn these cells reactive, it also augments the expression of this protein corrin 1A. And also it was validated in the brain sections of HIV-1 infected individuals. They too had augmented expression of CORIN1A. So for the next set of experiments, we sort of combined two different experiments, knockdown of our protein of interest and also introducing HIV-1 TAT into the cell system. So here, and then analyzing the astrocytic condition media, RNA, protein, and also taking this ACM and treating our human primary neurons with it and assessing their neuronal health. So in next experiment, I'll be showing you the results of the previous experimental setup. Here you can see that GFAP, which normally increases under the effect of HIV-1 TAT, gets rescued because of the knockdown of CORIN1A. And also, not only this, but release of IL-6, which is significantly enhanced under the effect of HIV-1 TAT, it also gets rescued, or you can say attenuated, when you downregulate CORIN1A. And also, there are some, genes and proteins involved in inflammation called as PTGS2, NOS2, VCAM1, ICAM1. There was rescue obtained at PTGS2 and NOS2 as well. So here you can see TAT enhances PTGS2 and NOS2 expression, but when you downregulate CORIN1A, that increase is not observed. So the levels are similar to the control levels. So in the next step, I took the conditioned media from those astrocytes and treated my neurons with it. And so I found that TAT, so a few more minutes, just one last minute. So HIV-1 TAT induces neuronal death, which was kind of rescued when you downregulate CORIN1A. Also, we found that HIV-1 TAT shows its effect. It augments CORIN1 expression by a microRNA called as microRNA92B5P, and mimic and inhibitor were employed to confirm its role. And so here's a summary slide which shows that when CORIN1A is downregulated, there is impairment in release of calcium, and probably that is the mechanism by which HIV TAT reduces 92B5P and increases CORIN1 expression and mediates glia-mediated neuronal death. So thank you, and I would like to thank NBRC Core and CSIR for the funding.
Thank you, Hriday.
Thank you so much.
So the last speaker for today's lightning talk is Sravanthi, a PhD student from TIFR Mumbai. Sravanthi, can you?
Yeah, I cannot unless you stop sharing.
Yeah, Hriday, can you stop sharing?
Yeah, thanks.
Yes, we can see your screen.
Yeah, thank you. I'll start now. Good evening, everyone. Today I'll be talking about the roles of LRRK2 and Liprin alpha in synaptic vesicle biogenesis and synaptic vesicle protein trafficking. Synaptic vesicles are important cargo which aid in neuronal function. Composition of synaptic vesicles found at the synapse is fairly well studied and understood, but information on early trafficking routes of synaptic vesicle proteins and synaptic vesicle biogenesis is still poor. We have previously shown roles for 3 genes, GIP3, LRRK2, and the AP3 complex in regulating early trafficking of synaptic vesicle proteins at the Golgi. We are now investigating roles for newer, more downstream molecules such as SIT2 or the C. elegans homologue of Liprin alpha. Liprin alpha is a synapse active zone protein and is also a known interactor of the synaptic vesicle motor UNC104 or KIF1A. Liprin alpha, LRRK2, and the AP3 complex have all been shown to affect lysosomal protein trafficking in different model systems. We therefore hypothesize, based on available literature, that there might be a post-Golgi sorting set, perhaps something resembling a synaptic vesicle lysosome intermediate, from which synaptic vesicle proteins are actively sorted into synaptic vesicle protein transport carriers. Through live imaging of synaptic vesicle proteins, through live imaging of co-transport of one synaptic vesicle protein, which is synaptogyrin1, and a lysosomal protein, CTNS1, we were able to identify the presence of an SV lysosome intermediate in C. elegans neuron. We see that in LRRK2 and AP3 mutants, these intermediates exit into the neuronal process, and they do so more frequently than in wild type or in Liprin alpha mutants. This suggests that the separation of lysosomal proteins from synaptic vesicle proteins early on in the cell body is mediated by LRRK2 and AP3, and that this early separation is perhaps critical for the biogenesis of synaptic vesicle protein transport carriers. We then asked if these genes act in the same pathway, and we see that in the doubles of liprin alpha with a LOC2 and AP3 complex, that there are fewer synaptic vesicle lysosome intermediates in the neuronal process. It is evident from this that liprin alpha and AP3 genetically interact to restrict the synaptic vesicle lysosome intermediate in the cell body, and that they perhaps act together and downstream of LOC2 to enable this separation. Moreover, we see that the mutants of LOC2, AP3, and liprin alpha have fewer vesicles in which they co-transport two different synaptic vesicle proteins, which is synaptobrevin1 and RAP3, suggesting that all these three mutants affect trafficking of synaptic vesicle proteins to synaptic vesicle protein transport carriers, thereby affecting the synaptic vesicle protein carrier biogenesis. These results suggest that the biogenesis of synaptic vesicle protein carriers is indeed a hierarchical process where LOC2 and AP3 act upstream of liprin alpha to first separate lysosomes from the synaptic vesicles, and then through liprin alpha to assemble and axonally target synaptic vesicle protein carriers of a defined composition. The misformed synaptic vesicle protein carriers in LOC2 and AP3 mutants mislocalize to the dendrite in an AP1 dependent manner. However, the misformed synaptic vesicle protein carriers retain their polarized distribution to the axon and liprin alpha mutants, suggesting that the loss in polarized distribution may be a consequence of severe trafficking defects which occur in LOC2 and AP3 mutants. Additionally, liprin alpha, like the AP1 complex, is able to suppress the synaptic vesicle protein mistrafficking to dendrites seen in LOC2 and AP3 mutants. In fact, we see that liprin alpha and AP1 complex behave similarly in that they do not mislocalize synaptic vesicles to the dendrites, but are able to suppress the mislocalization of synaptic vesicles seen in LOC2 and AP3 mutants. Moreover, the AP1 and liprin alpha double mutants drive synaptic vesicles into the dendrite, suggesting that they likely act together or redundantly to one another to restrict synaptic vesicle protein distribution to the axons. To summarize, we've identified a novel step in the biogenesis of synaptic vesicle protein carriers, which is separation of synaptic vesicles from a synaptic vesicle lysosome intermediate, mediated by LOC2 and the AP3 complex, which likely acts through liprin alpha. Once these two protein pools are separated, the synaptic vesicle proteins are trafficked to assemble a synaptic vesicle protein transport carrier by liprin alpha, likely under the regulation of LOC2 and AP3. The misformed synaptic vesicle protein carriers are driven to the dendrite in an AP1 dependent manner, perhaps again by liprin alpha. The correctly assembled synaptic vesicle protein carriers are targeted axonally and exit the neuronal cell body by recruiting KIF1A. With this, I would like to thank the organizers for the opportunity to speak here, and thank you all for your attention.
Thank you, Sravanthi. That was very interesting. And thank you all the speakers for the wonderful presentations. We'll have some more lightning talks tomorrow from our some more colleagues. We encourage all the speakers to answer the questions in the chat window and carry forward the discussions. So this ends the lightning talk session. And now I'd like to call Dr. Poonam Thakur. Over to you, Dr. Poonam Thakur.
Yes.
Can you take over for the next part of the program?
Yes, sure. So now we have come to the last leg of our program today. We will be having our next talk by Dr. Hiya Ghosh. Just a second. All right. So after Professor Torle taught us how to make a hippocampus, Dr. Hiya Ghosh is going to tell us how to maintain an adult brain. And before that, I'm so sorry. Please excuse. Yeah, so Dr. Hiya Ghosh is an assistant professor at NCBS, Bangalore, India. And Dr. Ghosh.
Yeah, hello.
Yeah, I'm so sorry about this technical issue. I hope yours is going better than this.
Yeah, it's I've been able to listen to all talks. It's been wonderful so far. Should I share my screen?
Yes, please go ahead. So yeah, I couldn't finish the introduction. Please allow me to do so.
Yeah, sure.
Yeah. So Dr. Hiya Ghosh is a recipient of Scholars Award from American Society of Hematology and also a recipient of a prestigious Ramanujam Fellowship from DST. Her lab works at the intersection of neuroscience, immunology, and genetics with a particular focus on brain homeostatic regulation and neuroinflammation. And we are very excited to have you, Dr. Hiya Ghosh, and over to you now.
Okay, so I like to start. Am I audible? And let me just put my laser pointer if it's visible. So things are set, I hope.
Yes, you're audible and visible.
All right. So, firstly, thank you so much to both Vasudha and Poonam for putting up this exciting symposia and giving me an opportunity to participate. It's really exciting the way the talks are organized and the students' participation, everything. I'm very excited to be a part of this. So the topic that I bring in today is maintaining order in the brain. Now, straight away jumping onto it, well, if it allows me to go. Yeah. So by maintaining order in the brain, what I mean is brain homeostasis. And as we all know that homeostasis is really the condition of optimal functioning for an organism, that is the steady state. And if homeostasis is successful, life continues, it's all good. But if it's unsuccessful, then dysfunctionalities happen, which can even lead to death. So what our interest is in to understand how do brain cells maintain their steady state. We all know that cells are regulated by intricate genetic program. And we are interested to find out what are the genetic programs that govern the steady state functioning of different brain cells. And the motivation here is that if we can understand what is enabling what kind of regulations enable the normal functioning of different brain cells, then perhaps we could better correlate pathological situations to deviation from homeostasis and then address them better. Now, with that, I'd like to start with this first part of my talk, which is to do with neural stem cells, but in the adult brain. Now, it's been about 5-6 decades now that we have known that neural stem cells reside in adult brain as well. And these cells look like this tree-like structure. They have a radial radiating process with a lot of branches at the end and a cell body. And these cells, however, are not there in all across in the parenchyma, but in very limited location in the adult brain. So this is a mouse brain. And new neurons have been found in the location of the hippocampus called dentate gyrus, and the other location has been olfactory bulb. Now I'll show you a coronal section of a mouse brain, wherein in this mouse, the stem cells have been genetically labeled with green fluorescence protein. And you can perhaps appreciate how few these cells are, only located in the dentate gyrus of the hippocampus and the lateral ventricles. And now if I zoom in the dentate gyrus, you can perhaps appreciate this blue hue, which is a pack of mature granule cells of the dentate gyrus and a thin layer of these green cells, which is the neurogenic niche in the adult hippocampus. And in the human brain, new neurons have been reported in the dentate gyrus and in the striatum. While dentate gyrus is common between rodent and human, the striatal neurogenesis is thought to be specific to the humans. Now, if we have stem cells in the brain, why do we have them? Why are neurons continually made? What are their functions? Well, to begin with, it is in the last 2 decades, many important findings have come through, mostly through experiments in rodents. And it has been shown that the new neurons forming in the adult brain actually participate in very specified type of cognition. So the first out of them would be pattern separation. What I'm showing you on this slide are 2 animals, very similar looking, sitting in a very similar posture. But I hope it would have been nice if I was interacting, but I'm hoping that most of us, if not all, are able to distinguish that these 2 animals are not the same, they are different animals. And we are able to do so because our neurons are encoding information in detail. And in doing so, it's able to keep them separated orthogonally. And this is allowed by a particular balance of excitation and inhibition of encoding. And this process is enabled by the new neurons that are formed continually in the dentate gyrus. The other function in which cognitive function in which new neurons of the adult brain are thought to participate is erasing memory. Believe it or not, forgetting actually helps us in our survival. If we forget, we have a chance to relearn something. And this is contextual in conditions such as fear memory. So if you go to a context where you have an unpleasant experience, what happens is that your memory encodes that context associated with that unpleasant experience. And therefore, every time you are back in that context, you are bound to be trapped by that unpleasant experience or anticipation of that. And in these cases, you'd rather prefer to not keep remembering that unpleasant experience because it's not necessary that every time that context will give you an unpleasant feeling. Now, in this experiment, this is a context, a particular cage, a specific type in looks and feel. In this, you put a mouse, and at the same time, you give it a food shock. So food shock is a very unpleasant experience. And so the mouse associates this context with that bad experience. Next time, if you bring the mouse in the same context, but do not give it any shock, it's likely to anticipate an unpleasant experience and freeze itself because it's coding, the encoding of association is still going on. And to show that the ability to reverse this learning depends on the formation of new neurons. An experiment was done wherein juvenile mice in which postnatal neurogenesis is seen to be much more than in adult mice were given a treatment of food shock. And alongside, an adult mouse was also given a food shock. And then they were brought back to the same context several days later and assessed for their memory by assessing freezing response. And as you can see, the adult mice seem to hold on to their bad memory to that context, whereas the new juvenile mice, where many more newborn neurons are born, rapidly forgets that unpleasant experience and perhaps is able to move on. Now, if you take the adult mouse and then give it an enriched environment which is known to enhance adult neurogenesis, then that same adult mouse also reduces its freezing response, showing reversal of memory. And finally, a third type of cognition is our ability to deal with stress. There have been many correlative examples which shows that environmental challenges which can cause stress, such as prenatal stress or early life stress, even social defeat, or artificially causing stress by glucocorticoid administration causes a reduction in adult neurogenesis. Conversely, ablating neurogenesis increases anxiety-like behavior in rodents and on the other hand, if neurogenesis is enhanced, it improves recovery from acute stressors. Also, it has been shown that adult hippocampal progenitors are reduced in cases of depression, and antidepressant treatments in major depressive disorder patients seem to increase the hippocampal or the dentate gyrus volume, which is where these new neurons are formed, indicating a possible increase in adult neurogenesis in response to antidepressant treatment. So with all this, one would want to know, can adult neurogenesis actually be manipulated? Can you increase or decrease it? Well, the answer really is good news. Yes, it can be altered, and it's been shown that it can be altered by simple enrichment of the environment. So in rodent experiment, if you keep mice in this boring normal cage with just bedding, there is a certain level of neurogenesis. But if you keep them in an enriched cage where lots of toys and wheels are there, then what you see is a dramatic increase in newborn neurons. This is the normal standard neurogenesis of adult mouse. And in case of enrichment, the newborn neurons are enhanced. On the contrast, if the mice experience a stress given by social isolation, then newborn neurons dramatically decrease. So I think what is clear here then, that neural stem cells of the adult brain help us to improve our cognition and also keep a good mood by enabling us come out of stress and anxiety. And this involves a process which can be manipulated through non-invasive environmental or physical changes. So this makes for a good case to understand more about the adult neural stem cells itself. So what is known so far is that the neural stem cells sitting in the adult brain can make astrocytes and can make neurons, and also can make oligodendrocytes if induced properly. What we asked is that all about the potential of the neural stem cells, or is there more to it? And our story really started with our observation that this transcription factor called TCF4, which is a BHLH transcription factor, already known to play an important role in fate determination in the immune system and also during brain development, was quite abundantly expressed in the neurogenic niche. So herein I'm showing you a part of the dentate gyrus. Previously, I showed you that there is a thin layer of neurogenic niche in the dentate gyrus. This is exactly that. And herein the neurogenic cells are genetically marked with green fluorescence protein. And if you see carefully, you will see that all these green cells express quite high level of TCF4. This neurogenic program in the hippocampus is very well characterized, wherein the quiescent stem cell is known to come out of its quiescence and go into rapid proliferation during its intermediate proliferating cell stage. And once it decides to take a neuronal fate, it differentiates to something called a neuroblast, which then goes on to mature and become a mature granule neuron. And each of these stages can be identified through the expression of a stage-specific proteins alongside their morphology. And using these information, we were able to actually demonstrate that TCF4 was being expressed in indeed every stage during this adult hippocampal neurogenic program. So this established that TCF4 must be important for adult neurogenesis. However, this also posed a challenge, because if we were interested to understand what TCF4 is doing specifically in stem cells, then we needed to find a way to only delete it only in the stem cells and perhaps the progenitor, but not the downstream progeny, and do all of this in adult brain. So for this, one could utilize this information that, for example, this gene continually expressed from the stem cell to the progenitor stage, and combine this with this powerful mouse genetic tool called LoxP-CREAR. I'm going to go into a little bit detail here for the benefit of the students. So I hope the senior people can bear with me here. So what we do in this tool, this is a powerful tool for manipulating the genome in mice. What you do here is you use a cell-specific promoter. For example, in our case, nestin, because it's expressed in the neural stem cells. And you use this promoter to express Cre recombinase, a bacterial enzyme, which recognizes specific sequences in the genome and recombines them, thereby deleting whatever comes in between those two sequences. You have this Cre expressed, but in a fusion format with the estrogen receptor to impart inducibility. And in the same mouse, what you also enable is design of these two LoxP sites. LoxP are the sites that are recognized by Cre, and flank them around the gene of your interest. Now you have and in our case, the gene of interest was TCF4. Now you have a cell where your gene of interest is flanked by the LoxP sites, and you're expressing Cre only in your cell type of choice, which is the neural stem cells here. And now the Cre is being expressed, but it's stuck in the nucleus. It cannot do anything until you induce its release by injecting estrogen mimetic, tamoxifen, which goes and binds to the estrogen receptor that results in release of the Cre, allowing it to go to the nucleus and cleave off recombine the LoxP site and cleave off the gene that you want to delete. What you get then is a gene deletion in a cell type of your choice and at the time that you wanted it deleted. Now to this, you can also add a genetic tracking system in which you add a GFP protein that is driven by a continually expressing promoter, but you put a stop cassette in between and flank it by LOXP side. However, when CRE is activated, just like the gene of interest, this stop cassette will also be removed and the protein will start getting made, making your knockout cell a green cell, and it stays green because the green is driven by a continuous promoter. And so you can actually now track this cell regardless of what happens to it except for death. So what you can do now is track the lineage of this cell to know what it becomes, and also purify a very purified deleted population to do further analysis through proteomics, transcriptomics, biochemistry, whatever else you want. So using this genetic deletion, which enabled us to specifically delete TCF4 in stem cells in the adult brain, we saw that immediately the number of newborn neurons in the hippocampus decreased dramatically. And so what happened? Through lineage tracking, and in the interest of time, I'm not showing all the data, it's already published. We first excluded the possibility of a fate diversion. We lineage tracked these deleted progenitors, and we did not see them become astrocytes or something else. But what indeed we found was a defect in proliferation, that these progenitors after TCF4 deletion were not amplifying properly to make newborn neurons. So the question then was, what happened to the progenitors as a result of TCF4 deletion? Now, if you remember the picture, the amount of stem cells in the dentate gyrus is extremely, extremely low. So to know more about what's happening to these knockout cells, which were already reduced in numbers, we had to culture these cells out from the mouse brain. So we microdissected the dentate gyrus and did something called a neurosphere culture. This is the established protocol because the stem cells have inherent property to clonally expand. And what that leads to is the formation of this tight cluster of clonally expanded cells called neurosphere. And when we did that in wild type versus in TCF4 knockout, the first thing we saw was that there were far fewer and smaller neurospheres in the knockout brain from the cells derived from the knockout. And most of these other cells did not have a neurosphere-like morphology. They were more flattened and had projected. So we suspected that they were probably losing their stem cell kind of nature. So to take another look at it, we performed something called a secondary neurosphere, which is nothing but you dissociate this clonally expanded tight ball and let them clonally expand further to make more neurospheres. And when we did that, it was immediately clear that TCF4 deletion had abolished the self-renewal property in these stem cells. So that is fine. It could not self-renew itself, but did something else also go on? What was the transcriptome? What else changed as a result of deleting this transcription factor? To understand that, we took these and did a transcriptomic study. And immediately what jumped out was this upregulation of these very weird looking genes, which are not to be mentioned in the entire neuroectodermal origin cells. So these genes, if you are familiar with the immune system, are mostly expressed in macrophages and had a more myeloid-like feel. And so we took all the upregulated genes and looked for their functional characteristic through an ontology analysis. And what it showed was, again, consistently with the expression of these immune-like cells, it shows immune response, cell adhesion, cytokine secretion, all things that sounded like an immune function. And since these cells look like myeloid in nature, we further did gene set enrichment matching. We took enriched gene sets for myeloid differentiation or macrophage-like genes and compared them with the upregulated gene signature. And indeed, the TCF4 knockout neurospheres or stem cells had more of myeloid-like gene signature than the neural stem cell signature. So this was the first evidence to show that possibly the neural stem cells were transforming into something that had a myeloid nature. Now, if that is really true, then what we would expect is that first thing, they'll not be able to make neurons because they probably are no longer neural stem cells. And secondly, if they are indeed myeloid inflammatory, then they should probably cause inflammation of some kind in their surroundings. So we tested these two. For the first one, we took these neural stem cells and put them in differentiation culture to make neurons. And as you can see in the wild type upper lane, the progenitors grew nice processes and stained for the neuronal marker TUJ1. Whereas in the knockout, to begin with, there were only very few progenitors. They did express TUJ1, but look at the morphology. It's very different. It's stocky, bushy cells that do not quite look like neurons. And consistent with our RNA-seq data, where MBP was one of the highest upregulated protein in the knockout cells, alongside TUJ1, these knockout differentiated cells also expressed the oligodendrocyte marker MBP. Alongside that, we also saw another myeloid marker, CD868, also showing up in the same cells along with the neuronal marker. And CD68 in the brain is exclusively expressed by macrophage, which is a myeloid lineage cell. So all in all, this neuronal differentiation shows that knockout of TCF4 in adult neural stem cells do not allow them to make normal neurons upon differentiation. What about inflammation? If it is that the myeloid transformation is real, then could we expect inflammation in the neurogenic niche? So to check that, what we decided to do was to go unbiased. We took the dentate gyrus, microdissected the dentate gyrus from the wild type and the knockout mouse brain, and we did transcriptomic analysis. And immediately what jumped out was that in all the three knockout brain, the top upregulated genes were lysozyme, were interferon responsive genes, showing a very uncharacteristic inflammatory property coming up in the dentate gyrus of the knockout brain only as a result of TCF4 depletion in neural stem cells. To further confirm that inflammation was indeed happening, we checked one of the hallmarks of parenchymal inflammation, which is microglial activation. Microglial activation is seen also by increase in CD68 expression in the microglial cell, and that's exactly what we see. So all in all, the study shows that the neural stem cells, which were thought to be only making good cells for the central nervous system, the neurons and the glia, actually have a latent myeloid inflammatory potential, which is continually kept suppressed through the expression of TCF4. And in the absence of TCF4, these stem cells cannot make neurons, causing reduction in neurogenesis. But not just that, it also causes inflammation in the niche, which could have further detrimental effect. So where does this lead us to with respect to brain homeostasis? Now, inflammation, I think you guys would agree, is a noted co-occurrence in many neurological disorders, whether it's psychiatric, developmental, and of course, neurodegenerative. And in most of these cases, whether inflammation is a cause or a consequence remain unknown. Now, with the revelation that the stem cells sitting deep inside the hippocampus have this detrimental potential, it will be important to ask whether neural stem cells could be a contributor in neuroinflammatory diseases. And this would be specifically relevant to diseases of hippocampal relevance, because that is where the adult neurogenic niche is. And these would be disorders such as stress and anxiety, neurodegenerative disorders, Alzheimer's in specific, and cognitive decline. And on the other hand, alongside the potential of adult neurogenesis, another exciting thing about the discovery of adult neural stem cell was in the context of regeneration. When we lose neurons, and if we have stem cells in the adult brain already, can we have it make for the lost neurons? So there is a lot of regenerative efforts that are focused around the stem cell. And in all these contexts, it would be important to watch out for this detrimental potential that we have just very unexpectedly revealed about adult neural stem cell. Now I'll change gears to the other important, very important cell of the brain, which is the mature neuron. And I think you guys, everyone will be with me if I say that mature neurons are perhaps the most complicated in structure cell of a body. And this complicated structure enables it for long range connectivity. And also they are kind of a little bit weird in the way that their functional units are dynamic, these spines that decorate the dendritic tree and keep coming and going as a functional dynamic unit of the cell. Alongside this, the mature neurons are also one of the longest living cells in our body. And yet they do not have much regenerative capacity. So one could legitimately ask what underlies, how is the structure and function of mature neurons maintained through a long lifespan, sometimes decades, in such as human beings. So in addressing that question also, TCF4 kind of came in handy for us, and that is because this is how TCF4 looks in the brain. It's expressed all across in the parenchyma. And when we specifically in the neurons, we were surprised to see that it was quite indiscriminately expressing both in inhibitory and in excitatory neurons. I hope you can see that TCF4 in this CAM kinase 2 alpha excitatory cells, as well as parabilmumin expressing inhibitory cells of the cortex. And this was not only true in the cortex, it was also true in the hippocampus, the striatum, and in the even in the cerebellum. And not only that, when we looked more carefully, and this is now established by other labs as well, TCF4 is expressed also in astrocytes, in oligodendrocytes, in microglia, and like I said, in neural stem cells. So this tells TCF4 is very important probably for understanding and can reveal a lot about brain homeostasis, but also poses a challenge that if you can learn more about brain cells' normal regulation by tweaking TCF4 only if you are able to manipulate it in a very cell-specific manner, and in our case, in the adult brain. So for that, I already explained that log-speed 3ER system, inducible and conditional deletion comes in very handy. And for studying the neurons, we exploited the CAM kinase 2 alpha expression, which is restricted to excitatory neurons. And inducing, what we did is we induced deletion of TCF4 in excitatory neurons in adult brain when the neuronal development was completed, the connectivity was all done. And then you go and delete TCF4 in mature neurons, what you see immediately is a profound change in morphology. Within 2-3 weeks, we saw that the whole dendritic structure of the mature neuron changed because we deleted TCF4 from it. And alongside that, we also saw that the spines, which are the functional structural unit on the dendritic tree, also had changed, indicating that probably there is something with activity as well. And sure enough, when we recorded from these cells, we saw that the TCF4 deleted mature neurons were far more excitable. For any given input of current, they showed more action potential, they fired more action potential. And consistent with that, we also saw membrane property changes, such as increase in resting membrane potential and a decrease in input current, input resistance. So this was very revealing, saying that TCF4 mediated gene regulation probably plays a critical role in maintaining both the structure and the activity of mature neuron. So the question is, what are these genes and pathways? Especially because TCF4 is a transcription factor, we probably can learn a lot more about what underlies structure and function if we know what are the genes and pathways. And this was not an easy deal to know, because when we looked carefully, now I've already explained the genetic tool that we use, which enables us to not only delete but also permanently label the deleted cells through the GFP expression. And so when we looked carefully, we saw that the CAM kinase 2 alpha expression mediated GFP expression labels only a certain percentage of the CA1 cells of the hippocampal neurons. And if you see carefully, there is DAPI everywhere. So this dark spot and the green spot both have pyramidal neurons, but only one expresses CAM kinase 2 alpha, the other doesn't. So just out of curiosity to know if these are different cells sitting next to each other and yet CA1 pyramidal neurons, we looked at the baseline activity by looking at CFOS, which is an activity marker. And quite remarkably, what we noticed that very few of the green cells actually had baseline CFOS positivity, whereas most of the CFOS was coming from the non-green cells. So this is further quantified and shown here to emphasize that the cells, two pyramidal neurons sitting next to each other in the hippocampus, may be completely different. They may have different baseline activity, and clearly they had different CAM kinase 2 alpha expression. And so the task was for us, we couldn't just micro dissect the CA1 from the wild type and the knockout brain and do a transcriptomic. We had to specifically isolate the green cells to really know what TCF4 does in a mature neuron. So for this, we went through the painstaking process of standardizing how to sort mature neurons from adult brain. First, you isolate, very careful isolation, and then you exclude the doublet cells, exclude microglia, and then from the remaining cells, as you can see, we could very clearly take out only the green neurons. And that is what we did a transcriptomic analysis for. And first thing is that, quite relievingly, there were only about 105 or 100 genes that were differentially regulated, out of which 90% of the genes were upregulated. Very, very few were downregulated. That immediately tells that TCF4 has a suppressive role in mature neurons. And amongst these genes that were upregulated, we found the things like aquaporine and ACE, which had no reference in the context of mature neuron activity or structure. So this was exciting also because they were completely unknown protein, although we had to go to a little more extent to confirm this. And when we did an ontology analysis, we saw that alongside functions such as plasma membrane and projections, we also noticed that cilium was coming again and again, no matter which stringent analysis we did. And indeed, if you look at the majority of genes that are influenced by TCF4 deletion, then the major cluster belong to genes that were previously known to function in ciliary pathway. And this is very exciting because cilia, even though it's thought to be present in every cell, its function and relevance in mature neurons, and especially its maintenance, is completely not documented. And just to be sure, because TCF4 is also known to be expressed, known to be important during brain development, we also compared it with its transcriptome in a P0 mice brain to see what it does in the developing brain. And that showed that in the developing brain, its main function, the cluster of genes that it regulates, has to do with mitosis and cell cycle. And therefore, clearly in the mature brain, it has a completely different set of functions, and it influences a completely different set of genes. So in conclusion, the study shows that TCF4 regulates critical gene modules that underlie both the structure and the activity of adult neuron. And where does it take us in terms of brain homeostasis? First thing is the revelation that ciliary genes may indeed be involved in mature neuronal maintenance. How and what these pathways, how they influence the structure and activity are something that we are pursuing on. Will be major interest to know all these new genes, aquaporine and ACE proteins that could influence neural activity, how they do it. But another important aspect of this study was the revelation that the mature neurons indeed harbor a proactive genetic program that allows it to morph its structure. And this is not fully surprising because there are evidences from pathology such as stress and aging, where postmortem samples of human brain has shown alterations in dendritic tree of mature in adult brain. But how this happens and what is the physiological relevance of these changes remain poorly understood. And we hypothesize that there could be a possible advantage at the circuit level if a neuron has the ability to morph, say in case it has to make up for the function of a neighboring neuron serving in the same circuit. So we are currently pursuing this. And overall, with these two studies, what we have been able to reveal are homeostatic regulations, some novel ones that underlie normal brain functioning, one in the context of neural stem cells and its potential to kind of modulate inflammatory situation in the parenchyma, and the other in the context of mature neurons and the possible participation of novel pathways involving serially genes in its activity and structure. So with that, I'd like to thank the most important people who did the work. I have got a great team of students. And this is Sharik, who contributed significantly to the first story of the adult neural stem cells. And Deepanita pretty much single-handedly led the mature neuron project. And finally, our funding agencies, which have helped us pursue what we want to investigate. So with that, I'm ready to take questions if there is time for that.
Yes, we do have some time for a couple of questions. In fact, we have got a lot of questions for you. It was a really, really fantastic talk. So I'll pose them one by one. So Shresh Shekhar from ICTBM is asking, what might be the mechanism for enrichment mediated increase in adult born neurons and more efficient pattern separation?
So I'm just trying to go to the chat box so I can also read. So could you repeat what was the question? Mechanism for enrichment mediated increase. Yeah. So
in the chat box it might be a bit mixed, so let me read it out to you.
Yeah, no, I got the question. I think the question was what mediates the enrichment mediated increase of adult neurogenesis, right?
Right.
Yeah, and this is $1 million question actually, because if you are able to understand how the non-invasive and potentially environmental changes, how these cues are induced, are mediated to the neural stem cells to take a decision, that would be hugely important and beneficial for all of us. So a lot of research is going on to understand how the environment is actually cued into the neural stem cells in the adult brain. And I will have to say that we do not understand a whole lot, except that we know that inflammatory factors from the circulation have been shown to adversely influence the neurogenesis capacity of the adult neurons. Oops, I think I have a load shading. Hello. Hello.
Yeah. So we have another question by Prakash Devaraju. He asks, will neuroinflammation affect TCF? Is it perhaps known that it will connect neuroinflammation and brain disorders?
So sorry, I had lost connection because my there was a electricity breakdown. So I think did I miss out on other questions? Sure.
Yeah. So Prakash Devaraju is asking, will neuroinflammation affect TCF? Is it perhaps known that it will connect neuroinflammation and brain disorders?
Yeah, so I think our study is the first one to reveal a connection of TCF4 and inflammatory transformation of adult neural stem cells. And we are obviously pursuing this to know now. So what we have done is a proof of principle to show that this potential exists. What we do not know is how physiologically relevant it is and in which physiological conditions would it manifest itself. So yes, the connection between TCF4, neuroinflammation, and brain disorders is what we are currently looking at by examining stress and anxiety model, as well as neurodegenerative models, specifically Alzheimer's and ALS, where adult neurogenesis had been documented to decrease.
Thank you. And maybe we'll take one more last question for want of time. So have you tried inducing TCF4? And if yes, did it improve the neurogenesis?
Yeah, so we have not. The answer is we have not. And I think that given that TCF4 is a suppressive, has a suppressive role, as you can see in mature neurons, it's very clear, but even in neural stem cells, the major amount of gene regulation is upregulated, shows it's a suppressor. So I would think that overexpression may not give a phenotype that you're looking for, because unless you are doing it in a physiological situation where neurogenesis is already decreased. What I mean is that there could be a saturation beyond which you cannot push the system. And what TCF4 seems to look like is a suppressive protein. So it would probably be more helpful to induce it in a physiological situation where it is decreased. and then you induce it to make up for the lost neurogenic function, perhaps. And we are basically now trying to search for the physiological relevance of when TCF4 induction could be relevant in neural stem cells.
All right, Dr. Ghosh, that was really fantastic. And we really thank you for participating in the symposium. I'm sure all the audience members have enjoyed the talk as much as we have. And we would now like to invite our last speaker of the day, Dr. Falguni Anand Alladi from NCAM2. And it's really a great pleasure for me to introduce her. She's a great scientist. She's a dear friend. She's working as a senior scientific officer at Department of Clinical Psychopharmacology and Neurotoxicology at NCAM2. And her lab is interested in molecular and cytomorphological mechanisms of how aging and age-related neurodegenerative diseases progress. And they have done some fantastic research on showing how Asian Indians have lower susceptibility to develop Parkinson's disease in comparison to Caucasians. She has been a recipient of many accolades. She's an elected member of National Academy of Medical Sciences. She's a winner of Professor A.M. Namasivayam Award from Indian Association of Biomedical Scientists. And she has also been a visiting professor at Goethe University in Frankfurt, Germany. So we would welcome you, Dr. Falguni. You may please start the presentation. Thank you.
Poonam, can you please permit me to share my screen?
We can already see your screen.
Is it full screen?
No, not yet.
Is that right?
Yes, now it's perfect. It's full screen and we can see you and hear you.
Okay, so at the outset, I'd like to thank Poonam and Vasudha for giving me this excellent opportunity to interact with the audience, and I'd be happy to answer any question which arises during the discussion or at the end of it. So what I would be discussing, as Poonam mentioned, is that our two windows to explore the neurobiological basis of etiopathogenesis of Parkinson's disease. So this is the building where which houses my laboratory, and these are the two windows that kind of ventilate my lab, both intellectually as well as theoretically speaking. Yeah, so as we all know, I mean, I'm going to share some basic aspects of Parkinson's disease just for the sake of the freshers and people who are not very familiar with Parkinson's disease, etiology, and the general specifics. So as most of us know, Parkinson's disease is the second most common neurodegenerative disorder. And it's a disease of the elderly, because the median age of onset is around 60 years of age. And more than 10 million people are currently known to be suffering from this disease as per the estimates given by the WHO as well as the Parkinson's disease societies that are studying or facilitating the studies of Parkinson's disease. The estimated crude prevalence, that is the number of people who are at any given time suffering from, have been identified to be suffering from Parkinson's disease is around 260 per 100,000. So this is generally derived from door-to-door studies which are conducted by specialists, movement disorder specialists, and they arrive at a number after examining at least a population of 100,000. The incidence is around 16 to 19 per 100,000. That is the number of new cases that come up in a population, in such a population within a year. What's interesting is that the prevalence of Parkinson's disease differs significantly between different populations of the world based on the ethnicity, the genetics, and the environment. Now, we all know that Parkinson's disease has been described in ancient medicine. Particularly in India, it was known as Kampavata, which was described maybe around 5,000 years ago. It has been mentioned in the ancient text, which is known as Charaka Samhita. And we also know that the seeds of the Mucuna pruriens are the natural source of levodopa, and it is interestingly in clinical trials currently. Similar texts from the Chinese origin also mention the occurrence of Parkinson's disease, and that traditional medicine is currently still in use and is likely to go into, again, clinical trials. And there is definitely a mention of the occurrence of Parkinson's disease in other ethnicities and other countries. The traditional text of other countries like the Greek population or the other European populations as well. Now, when we come to the modern medicine, we all know that Parkinson's disease was first described as shaking palsy by Parkinson himself and James Parkinson himself, and that was kind of 200 years ago. However, the name, sorry about that. Yeah, so this picture shows the place where James Parkinson was living, and he was, in addition to being a medical specialist, he was also a paleontologist. Certain aspects of so in addition to being, am I audible, Poonam? Is there a lag?
We can hear you fine.
Okay, right. So yeah, so as I mentioned earlier, James Parkinson was responsible for identifying this in modern medicine. And if you look for literature and Google search, if you look for the picture of James Parkinson, this is the picture which comes up more frequently. And I must mention over here that this is not the picture of the real James Parkinson. This is actually, unfortunately, his picture is not really available. So the credit for giving the name Parkinson's disease goes to the well-known neurologist, Jean-Martin Charcot, who described the importance of malady de Parkinson's or Parkinson's disease. He is also attributed to various other neurological identification of various other neurological disorders like the multiple sclerosis or amyotrophic lateral sclerosis, the Charcot's joint or Charcot-Marie tooth disease. And basically, he from while gathering the history, patient's history, he realized that the symptoms that manifest are seen in many of the patients, then they are in reality nothing but the cry from the suffering organs. So he basically highlighted the importance of history taking and the details that must go in to diagnosing any disease. While looking at the anatomy and pathology of Parkinson's disease, we know that the basal ganglia, which are majorly affected in Parkinson's disease, are a cluster of neural clusters, basically comprising of the caudate nucleus and the putamen, that is the striatum. It is separated by the internal capsule. The globus pallidus interna and externa also participate in the pathology of Parkinson's disease, and so do the subthalamic nuclei. In addition, the midbrain plays an important role in the pathology of Parkinson's disease because here are housed the dopaminergic neurons of the midbrain. That is, these are the neurons that throw the dopaminergic connections to the caudate and the putamen. So in a normal condition, this is the midbrain photograph of a midbrain of a normal individual, where you can see the black pigmentation And this is because of the presence of neuromelanin in the dopaminergic neurons. Now, since the dopaminergic neurons are lost in Parkinson's disease, there is an effect of, there's an effect called depigmentation, which leads to the pallor that is seen in the midbrain of the Parkinson's disease patients. Now, these neurons in the substantia nigra pars compacta are known to harbor alpha-synuclein positive Levy bodies, which form the neuropathological hallmark of the disease. So the reduction in the number of dopaminergic neurons and the dopaminergic innervation to the striatum leads to the prominent radiological findings, which are generally seen on PET MR. And as you see over here, this is a normal striatum of a normal individual, whereas this is from the Parkinson's disease patient, and you see there is a reduction in the luminosity on one side, and this is known as asymmetric loss of innervation, dopaminergic innervation, and this is typically seen in Parkinson's disease cases. Now, there is some kind of method to the madness, because if you look at the topographical progression of the disease, one can see that much before the onset of the loss of dopaminergic neurons and the associated motor disturbances, the prodromal features like constipation, anosmia are seen much ahead of the motor symptoms, that is roughly 20 years ahead of the onset of the motor disturbances. And the anosmia and constipation is often followed by sleep disorders, or sleep disorders like REM behavioral disorders are extremely important prodromal features that are seen in Parkinson's disease patients. Now, if we were to count the number of risk factors that are associated with this disease, one would see that rural living, well water ingestion, and environmental toxin exposure, which kind of gel well together, are involved in as are major risk factors for the occurrence of this disease. In addition, there are other factors like the middle age, obesity, lack of exercise, or genetic inheritance, which is seen in 10 to 15% of the cases of Parkinson's disease, are important aspects. What interests us are the age and the gender associated aspects of Parkinson's disease, as well as ethnicity. And I'll come in, I'll discuss about these in detail in a couple of, after a couple of slides. So we all know that there are 15% of the cases of Parkinson's disease generally manifest at an earlier age. That's less than 45 years of age, whereas remaining 85 to 90% of the cases that manifest later, that is after 60 years of age. And so the earlier onset is generally associated with a monogenic cause. That is, there would be a mutation in one of the known genes like Parkin, PINK1, DJ1, Lhg2, and the alpha-synuclein. Whereas in sporadic cases, also certain genes have been known to be implicated, like the Lhg2, the Parkin, the PINK1, and DJ1. The importance of alpha-synuclein has been rightly highlighted by the team of Professor Brack, where they hypothesized that the progression of the pathology of Parkinson's disease has a particular character. That is it probably begins somewhere in the periphery and it progresses towards the central nervous system. Right. So what did Brack suggest? So his team suggested that in the pre-symptomatic phase, you do not see any alpha-synuclein pathology in the regions that are generally associated with the motor disturbances. Say here, if we see the pathology begins in the dorsal motor nucleus of the vagus, then proceeds towards the locus coeruleus, and then in the third stage, it reaches the substantia nigra pars compacta. As we all know, this is the region which houses the dopaminergic neurons. Thereafter, it progresses towards the cortical areas, the mesocortex, the neocortex, and the secondary and primary neocortical association areas. So what he also suggested was that there's some kind of a dual hit hypothesis, wherein the first hit would be from the periphery, say either from the olfactory bulb or from the duodenum or the pancreas, and then it progresses by the vagal nerve into the central nervous system. Well, this has been documented or evidenced by several autopsy studies, particularly the first one, which came from Jeffrey Cordova's group, where they have shown that the grafts that were derived from post-commissural putamen of a patient who was grafted with a fetal nigral transplant, and after 14 years, when the autopsy tissues were studied for alpha-synuclein expression, they found that the alpha-synuclein expression was found even in the fetal transplant. That means the host alpha-synuclein, which was pathogenic in nature, had migrated from the host cells to the fetal cells which were transplanted. So that means there is a neuron-to-neuron transmission of alpha-synuclein. Coming to the other two risk factors, one is age and the other is gender. So as is seen in this slide, one can see that there is enhanced risk in the male, and there is an enhanced risk of Parkinson's disease with increasing age. So now that brings us to our first hypothesis, or rather our first query. We wanted to understand what is the role of ethnicity in the pathogenesis of Parkinson's disease. Now, if you see this world map, which is represented in different colors, so here we can see that the prevalence of Parkinson's disease differs with different countries. This is the lowest and this is the highest. Now, this map, again, as we can see, shows that the prevalence of Parkinson's disease is much higher in the Caucasian population compared to the non-whites of Africa, of China, of India, and so on and so forth. So these differences have been calculated based on certain studies which have been conducted in these different continents. Say, if you look at this particular review, Pringsheim et al. Show that in Asia, the prevalence of Parkinson's disease is much less compared to those, the Caucasians, which are Caucasians from the Europe as well as the South America. We have certain numbers even from the Indian context. So in the Indian continent, if we can look at the studies which have been conducted in early 1980s to recent studies as well, we see that the crude prevalence rate is much less compared to what was reported in the Caucasians, other than the Parsis of the Mumbai area. So we all know that Parsis belong to a different genetic population. I mean, they have a different genetic makeup than the Asian Indians, and so it's expected that they have slight differences in their in the prevalence of Parkinson's disease in this population. Nevertheless, what is important is that Asian Indians in general have a much lower prevalence of Parkinson's disease compared to Caucasians. So now this particular question intrigued a neurologist from NIMHANS, that's Dr. Uday Muthane. He and a team from UK Brain Bank, they conducted a study and looked at the number of melanized nigral neurons with increasing age. And for this, they looked at the normal human brains from an Indian brain bank as well as from the UK Brain Bank. And what they found was that the number of nigral neurons was consistent, was preserved in the Asian Indians compared to the loss of nigral neurons that was seen in the UK Brain Bank, the midbrains that were procured from the UK brains. So there was a distinct difference between the number of nigral neurons that were seen in the Asian Indians as well as the Caucasians. Now, the question which automatically arises is, what are the neurobiological factors that govern these differences? So what we did was, the first thing which one must do while comparing populations is that you have to standardize the techniques that are used. So what we did was, we followed a certain protocol, which was being followed by different Caucasian, different studies that have been reported in Caucasian populations. And one such study was from the Rush Presbyterian Hospital at Chicago. And what we found was that with age, as we can see over here, there was no actual loss of dopaminergic neurons. These are the Nissl stained cells, and there was no loss of dopaminergic neurons with age in our population. So the next question which we asked was, is this a phenomenon which is seen only in the Indian cohort, or are there any other studies which have focused on such investigations on age-related cell loss? And what we found was that in the Caucasian populations, like that from the Finland or the Denmark or USA, indeed there was an age-related cell loss, whereas in the French population, there was no age-related cell loss. Now, when we revisited the epidemiology aspect of the French population, we found that amongst the Europeans, the French people had the least prevalence of Parkinson's disease. So it was not surprising that age-related cell loss was an important aspect that was to be taken into consideration. So now, since we had standardized our techniques, our estimation protocols, we moved on to look at the neurobiological factors. And what we first looked at was the GDNF receptors, because we know that these neurons are GDNF responsive, and it is an important aspect for regenerative therapies as well. And what we found was that, again, there is no loss of GDNF receptors and the receptor-bearing neurons in the Asian Indian population, that is our cohort. The next important aspect, as we all know, is the alpha-synuclein aggregation to form the Levy bodies, and which can be observed by either H&E staining or ubiquitin immunoreactivity and alpha-synuclein immunoreactivity as well. This can also be seen by ultrastructural analysis. And what we found was that in our cohort, we found these Levy body-like structures only in the elderly brains, that is above 69 or 70 years of age. Now, when we counted the number of alpha-synuclein immunoreactive neurons in our cohort, we found that they increased slightly with age, but they were not as logarithmic as that is reported in the Caucasian population. So that showed that there's some kind of cytoplasmic trigger which goes on at a later age in our cohort. However, there indeed were ubiquitin positive Marinesco bodies which we saw, and they increased with age. So what we proposed was that there's some kind of subthreshold neurodegeneration that occurs in the brains of the Asian Indians. So the next question, so thus far we had looked at the neurons, and the next question which we asked was what happens to glia, because glia are involved in the aging phenomenon in the human brain. So to our surprise, we found that there was no major increase in the number of astrocytes or microglia, but there was kind of a morphological alteration. So in the younger brain specimens, we saw that the astrocytes had these slender processes, very little staining in the cell bodies, but with increasing age, the processes became stubbier. There was an increase in GFAP expression, and a similar morphological transformation was seen in Ldb1 positive microglia. So where we saw several chain forming microglia, as well as, so they probably could be the migrating microglia, as well as the phagocytotic microglia. However, there were no major changes in the CNPase immunoreactive oligodendroglia, as well as the calcium binding protein S100 beta. So to summarize our findings on the human tissues thus far, In the Asian Indians, we saw that the number of nigral neurons were more compared to the Caucasians. The neuronal loss, age-related neuronal loss, was absent in our population, but it has been reported to be 5 to 7% per decade in the Caucasians by other groups. And there was no age-related loss of the dopaminergic markers like the tyrosine hydroxylase or NER1 with age, and the GDNF as well as its receptors were preserved. However, there was an increase in the Marinesco bodies and a small increase in alpha-synuclein as well. The incidental Levy bodies were lowest in our population, much higher in the Caucasian populations, and the glial changes were negligible. But quantitative changes was negligible, but yes, indeed, there were morphological changes suggesting that there is some kind of subthreshold neurodegeneration that is taking place in the brains, midbrains of the Asian Indians. Now, unfortunately, there are no studies in the Caucasian populations with respect to glia and the changes in the substantia nigra pars compacta, but we did not, we also did not have human tissues for direct comparisons. So now we break at that and we change our gears again, and we come to another important aspect of the human life globally, that is admixing of populations. And as we can see, these arrows that have been indicated are basically talking about the admixing of populations, say, like in the Indian context, what is important is about the Anglo-Indians. And as we all know that Anglo-Indians, according to the Constitution of India, are the first generation progeny of people, of males who were of the English or the European origin or European descent, and the female was from the Indian origin, and they were domiciled in the territory of India. And these are many of the prominent community members and whom all of us know. So what is interesting is that an epidemiology-based study on the prevalence of Parkinson's disease in this community revealed that in the Anglo-Indians, the prevalence was 5 times lower than even the Indians. So that suggested that there's some kind of, there's probably some transmission of the neuroprotective factors from the Indian mothers, which has been transmitted or has been amplified in the F1 progeny. So as I mentioned earlier, one is we did not have tissues from the Caucasian population, human tissues from the Caucasian population for direct comparison. The second thing is we did not have human tissues from the Anglo-Indian community because of ethical concerns. So we then decided to design some models to understand the phenomenon, this particular phenomenon. And we thought of certain neurotoxin-based models, like we all know that rotenone is a very prominent molecule, which, or rather neurotoxin that has been used to study the progression of Parkinson's disease. It induces degeneration of subset of nigrostriatal dopaminergic neurons, and it leads to the development of Parkinsonian features like the motor behavior. So is 6-hydroxydopamine, which accumulates again in the cytoplasm, produces reactive oxygen species, and again causes progressive degeneration, which is seen typically in the Parkinson's disease cases. We selected MPTP as a neurotoxin of interest. And as we all know that this is a meperidine molecule, it enters the blood-brain, it crosses the blood-brain barrier and is taken up by the glial cells, converted by the resident MAO-B enzyme into MPP+, which is then released into the niche and which is taken up by the striatal terminals of the dopaminergic neurons. And then it affects the mitochondrial complexes, the synaptic vesicles, and several enzymes, ultimately leading to the death of the dopaminergic neurons. So now we'll quickly recollect what we have seen in terms of role of ethnicity in Parkinson's disease. We've seen that Caucasians have a high prevalence of Parkinson's disease, lower number of nigral neurons, whereas the Asian Indians have lower prevalence of Parkinson's disease and higher number of nigral neurons. And we were interested in looking at the Anglo-Indians, who have a low prevalence of Parkinson's disease, and now we need to know what happens to their the dopaminergic source and the dopaminergic terminus. So what we did was, using the MPTP model, which is generally used in association with C57BL6 mice, which shows a much higher susceptibility to MPTP because it has lower number of nigral neurons. And we chose another interesting mouse strain, which is the CD1 white mouse, which has interestingly much lower susceptibility to MPTP, have higher number of nigral neurons, and then we cross them to understand these molecular features. And so this brings us to our observations. Here, this is the C57BL6 mouse, this is the CD1, and these are the crossbred F1 progeny, which in our case represent the Anglo-Indians. So the C57 represents the Caucasians, and these are representative of the Asian Indians. And as we can see here, the number of dopaminergic neurons are much higher, again, even in the crossbreds. Similar observations were seen in the different attributes of the substantia nigra, that's its volume, the neuronal density. And after MPTP injection, to our surprise, we saw that yes, indeed, there was a focal loss of dopaminergic neurons in the C57BL6 in the lateral aspect of the substantia nigra, whereas in the CD1, there was just around a 15 to 17% loss. However, surprisingly, the crossbred showed no loss of dopaminergic neurons, and this was confirmed by quantification of apoptotic neurons using the tunnel technique, and we found that there was a much higher number of neurons which were actually undergoing cell death even at adulthood compared to the crossbreds, which showed much lesser expression. And this was complemented by the expression of the apoptotic markers like the caspase-3 and apoptosis-inducing factor, where we found that after MPTP injection, the expression of both caspase-3 and AIF was indeed reduced in the crossbreds. So that was kind of interesting, because that meant that there's some kind of upping of the neuroprotective potential in the striatum, which is the source of GDNF. And indeed, there was an increase in the expression of GDNF in the striatum of the MPTP-treated F1 and X2 mice, that is the crossbred progenies, and which was also reflected in terms of tyrosine hydroxylase expression and the volume of the striatum. So then the next aspect that we investigated was the functional aspect of the striatal neurons, and here we saw that the power of the local field potentials was slightly higher in the crossbred progenies, and post MPTP, this was the LFPs were completely deregulated in the parent strains, that is the C57 and CD1, but were much better preserved in the crossbred strains. Now, the other aspect, the aspect of neurotoxicity in the dopaminergic neurons is the level of calcium binding proteins that are present within the dopaminergic cells, because in response to any excitotoxic insult, there has to be calcium sequestering. And what we found was that in the dopaminergic neurons of the crossbreds, there was an upregulation of the calcium binding protein, suggesting that there's some kind of neuroprotection offered by the calcium binding protein. And this was evident in the behavior, where we saw that in the C57 showed an earlier latency to fall compared to the other parent strain, that is the CD1 and the crossbreds, and similar observations were seen in the grip strength that of the forelimbs. So now we know that MPTP generally affects the complex 1 and other mitochondrial complexes, more importantly the complex 1. So we studied the responses of complex 1 and complex 4 in in these mice strains, and we found that the complex 1 was downregulated in the C57BL6 and in F1X1, where the mother was of C57 line, and we saw there was kind of a compensatory increase in complex 4. So the next question we asked was what happens to the fission and fusion associated.
Dr. Falguni, sorry to interrupt. Can we request you to wind up in next couple of minutes, so we have still some time for questions?
Sure. So what we found was that there was an increase in the fission associated proteins in the susceptible strain. And serendipity is that the mitochondria of the astrocytes in response to MPTP showed an enlargement and stability and a structural stability. We also found that these differences were actually prominent from the beginning, that is and probably suggesting that the susceptibility to PD actually may arise during the ontogeny of the midbrain dopaminergic neurons. So the next aspect has a clinical association. We were interested in understanding the biomarkers of Parkinson's disease, particularly because the Indian context we see it has a different disease manifestation. It has earlier onset and a prolonged prodrome. So here we went back to the literature and we found that there were no consistent findings in terms of either A-beta or total tau or phosphorylated tau when we looked at the Parkinson's disease with cognitive impairment. So we performed some non-targeted biomarker studies using proteomics as a modality, and we found that 42 proteins were differentially altered in the CSF of our patients, and most importantly, the fibrinogen molecules were much upregulated. As we can see, there was a 9 and 7-fold increase in these proteins. So what we did was we used our same animal model, that is the C57BL6. We injected intraperitoneally, and we conducted some behavioral analysis, then novel object recognition test, the pole test, and what we found was that indeed fibrinogen and another molecule, CFH, were independently able to induce motor and cognitive deficits. So fibrinogen and CFH are likely to be good biomarkers for the disease in our population. So the findings thus far, to summarize them, we found that there's threshold neurodegeneration in the midbrains of the Asian Indians. Using the mouse model, we found that there are ethnicity-based genetic compositions that can heighten the susceptibility to Parkinson's disease. So it is not necessarily associated with major mutations, but it is yet governed in a multifactorial manner. There is a critical ratio of neuron to interneuron to glia, which is important for CNS functions, and fibrinogen and CFH can be putative biomarkers. The current leads that we have are on a role of glia and organellar involvement. So I would like to thank my team of students as well as postdocs. Here he's Vidyadhara and Afang, who established the admixing model of Parkinson's disease. Aditi worked on the biomarker aspect. Pilash worked on the glia. Jyoti worked on the glia in the human context. Vidisha is looking at the organellar story. Our studies have been funded by several funding agencies, government funding agencies of our country. And I'd like to thank my collaborators, Dr. Shankar, Dr. Mahadevan, Dr. Muthane, Dr. Raju, and Dr. Bindukuti. So I'd also like to leave you with a philosophical thought about the windows that I mentioned in my the initial or rather in the title of my talk. So and with the question that which area in the brain would be activated if a person is sitting and staring at a window deep in thought and introspection. I also invite the people from the rest of our country and the world to visit Bangalore. Bangalore Palace has beautiful windows as well. Thank you.
Thank you so much for the enriching talk, and we have lots of questions for you. So we will take some of them.
Yeah, I'm sorry there was some kind of shiver from my side because of the internet connectivity. Apologize for that. Yeah, thank you.
No problems. You could hear you absolutely fine throughout the talk.
Thank you.
So Ranjita has a question. What is the sample size of the quantitative and morphological study of glia for the young and aging population?
Okay, so in each, we had, in total, we studied around 40 brains, midbrains. And so for the young population, we had around 5 to 7 brains less than 10 years, and up to 30 years, we had another 10 brains. So coming to around 20 brains, midbrains. And I'd like to clarify that this is serology-based investigation. So where we slice the midbrain, human midbrain, into around 200 sections, and use principles of serology to count the number of neurons as well as number of glia and any of the profiles that we studied.
All right. So we have another one from Anagha Murlidharan. She asked, from our data on Anglo-Indians and the gender trends of Parkinson's, it seems that females have some preventive factors. What is the likely source for this factor? Is there a possibility of an increase in this factor in female progeny?
Yeah, indeed. It's a very interesting question, because I think many of the neurodegenerative diseases that, which have been covered by epidemiology, find similar observations of reduced prevalence in females, other than Alzheimer's disease. So the reason is almost invariably associated with estrogen and the hormones that are associated, that are exclusively found in women. Yes.
So Prakash Devaraju has another comment or question that is kind of in continuation of that. So the comparison between Indian and Anglo-Indians is quite interesting, considering the mitochondrial pathology. So mitochondrial DNA from mothers may be a protecting factor.
Indeed, yes, and that is a possibility, and we really need to explore that in detail, because we find these differences even at the ultra-structural level. Yes, it would be really interesting to study in future.
Yeah, so he already has a follow-up question on that. So if you could elaborate on the neuronal versus astrocytic mitochondria differences that you have observed.
So can I go back to that slide?
Yeah, please go ahead.
Just a brief. Yeah. So basically, this is a control C57BL6, and this is after MPTP. And as you see, there is a shrinkage of the mitochondria after following MPTP. However, in case of CD1, you find that the mitochondria have slightly shrunken, but the electron density is maintained, which is not the case in C57BL6. Now this is the neuron story. Here you can see again at higher magnification, the mitochondria are fairly well maintained, whereas the ER is dilated. It's dilated, not fragmented, as seen in the C57BL6. Now when we look at the astrocytes, we see that post MPTP, the size of the astrocytes has increased. So there was an elongation of astrocytic mitochondria, and it is believed that elongated mitochondria have a better calcium quenching capacity. So it is likely that it is, this is self-propagating, because as is known that even astrocytes respond to alpha-synuclein and the other Parkinsonian toxins. So it is possible that astrocytes either are able to withstand these neurotoxins because they have higher amount of monoamine oxidase B, which is absent in the neurons, and hence they have a different the mitochondria of astrocytes have a different response. The other possibility is that this helps in inducing, or rather in the self-propagation of astrocytes, or inducing gliosis that is seen in the disease. So it's very important to understand the pathological response of astrocytic mitochondria. Also, because if we look at the cytochrome oxidases or response to coenzyme Q10, which has been used as a therapeutic, has been used as in the therapy. As a therapeutic modality for Parkinson's disease, and we have seen it has failed at clinical during the clinical trials. Phase 3 of clinical trials. So these differences in the astrocytic mitochondria could play a key role. Thank you.
So we will take one last question, although we have multiple. So Garvit Bansal has a number of questions. I'll take one of them. Which is why you didn't take out human stem cell and differentiate them in lab to study them?
Sure. I think one of my colleagues, and she's also a great collaborator, Dr. Indrani Datta, is working on human stem cells. And this is going to be a prospective collaboration, prospective study. Yes. Thank you.
All right, Dr. Falguni. So we once again thank you for joining us despite your injury and giving us such a long, history of Parkinson's disease and some of the fantastic work. And with that, we have come to end of today's sessions.
Pleasure is entirely mine. Thank you, Poonam. Thanks again.
Thank you. Thank you so much.
Sorry to interrupt you. Yeah.
No, please. So tomorrow we will continue with several more of interesting talks. We are going to have talks by Dr. Rishikesh Narayanan, Dr. Nixon Abraham, and we will also have some of our young colleagues giving another set of lightning talks tomorrow. Winners of the best lightning talks will be announced tomorrow. So we request all the speakers of lightning talks of today to also be present for that ceremony. So when they are there, when the prizes are announced. And the day will be closed by talks from Dr. Soumya Iyengar and Dr. J. Gopalakrishnan. So thank you all for your enthusiastic participation and great questions. We thank all the speakers as well for joining us. And we look forward to welcome you on day 2 of Synapse Symposium. Thank you. Good night.